Good morning, everyone, and welcome to the ABTA Advances in Brain Tumor Research and Clinical Trials Patient and Family Meeting. My name is Emily Lippert, Program Manager here at the American Brain Tumor Association. We are delighted that you have joined us online today. The ABTA's patient and family meetings are free events typically held across the U.S. where brain tumor patients and loved ones can network and hear from experts about the latest updates in brain tumor research, treatment, and care. We chose to host our meetings virtually the last few years to protect the health of our community during the COVID-19 pandemic. Although we are saddened we cannot connect and network in person, we hope our virtual program will be an enriching experience with you, and we hope to see you in person soon. Since we are unable to meet in person, we thought a good way to connect and learn about our attendees who are joining us today is to participate in some polling. When a poll is launched, a box will pop up prompting you to respond to the question. Your answer will be anonymous and only collective results will be shown. Let's launch our first poll now. All right, our first poll today is, what region of the US are you tuning in from? Now, if you reside outside of the US, there's an option here that says international or outside the US. All right, results are coming in. We have every representation except for the Southwest right now. Oh, we finally got another response for Southwest. Keep them coming. Closing in 3, 2, 1. All right, so we have the winner with 46% from the Midwest. It's great to see that we have representation from all over. All right, let's launch our second poll. So please rate your overall understanding of research and clinical trials. Today, we will be talking all about research and clinical trials. So this will give us an understanding of where you are at pre-presentations and then where you will at post-presentations, where we will launch the poll again. Keep responding. We still have a few more trickling in. 3, 2, 1. All right, so we have 59% with a general understanding. That's actually a pretty good amount. We really do hope to see a great and excellent understanding after the event in hopes that each of you will learn something today. All right, great job, everyone. We now know a little bit more about you and our audience members who are joining us today, and we hope after today's meeting, each of you will gain new information. I would now like to thank ABTA's Program Planning Chair, Dr. Michael Vogelbaum, for his leadership in the development of this excellent program. He's joining us with me here today, and I just want to share a little bit about him before he says a few words. Dr. Vogelbaum is Program Leader of Neuro-Oncology and Chief of Neurosurgery at Moffitt Cancer Center and Professor of Oncological Sciences at the University of South Florida Morsani Medical School. In addition to his active neurosurgery and radiosurgery practice, Dr. Vogelbaum has been primary investigator of numerous local and national clinical trials of new drugs and surgical techniques and devices for brain tumors, which makes him a perfect candidate for today's program. He is the founder of the Reno Response Assessment and Neuro-Oncology Group, co-chair of the Brain Committee for NRG Oncology, and vice president of the Society for Neuro-Oncology. He has an externally funded, including NIH R01 funded basic science and translational research program, which currently focuses on immunobiology of brain tumors in various aspects of drug delivery to brain tumors, including both phase 0, window of opportunity clinical trials, and trials that involve active delivery of therapeutic agents to the brain. The ABTA is truly thankful for you, Dr. Vogelbaum, for your leadership and preparation for today's program and all the lineup of speakers and topics. Please say a few words for us today.
Well, thank you so much, Emily, and thanks to the whole ABTA team, and especially Mindy and Embreen for helping to organize and run today's program. Today, we're fortunate to have a panel of expert physicians and scientists who have devoted their careers to helping patients with brain tumors. Each one of them has made meaningful contributions to better understand how these tumors arise and develop, avoid our body's defenses against cancer, and resist our conventional treatments. They've also engaged in the development of new therapies, many of which have been evaluated in the setting of clinical trials. For today's program, we have challenged our experts to describe the whys and hows that underlie the scientific approach to determining the biological underpinnings of brain tumors and to develop more effective and patient-friendly treatments. You'll also hear from a patient whose life was changed as a result of a clinical trial that resulted in control of his very deadly brain cancer. Throughout the program, you'll have the opportunity to ask our experts questions, and there will be a roundtable at the end of the program where our experts will address many of the challenges they face in launching and successfully completing the clinical trials that are absolutely needed to advance new treatments. Thank you for joining us this morning. We look forward to providing you with an informative and hopefully engaging program. Emily?
Thank you so much, Dr. Vogelbaum. That was a great overview. I would also like to thank all of our speakers for taking the time out of their day to share their insight and expertise. We truly are looking forward to each of the presentations. Additionally, thank you to ABTA's corporate partners, Novacure and GT Medical Technologies, for their support of this virtual event. Without the time and effort from each of these individuals and organizations, we would not be able to bring you this quality educational programming that has brought together that brain tumor community. All right, so let's begin our first session. Our first session of today's meeting is all about the American Brain Tumor Association. The ABTA continues to be a proud advocate, and we are steadfast in our mission to be a strong ally and resource for the brain tumor community. This presentation will discuss the many ways that we are here to serve you and how you can get more involved. Please help me to welcome ABTA's President and CEO, Ralph DeVito.
Good morning, Emily, and thank you. And let me say on behalf of the volunteers and staff of the American Brain Tumor Association how much we appreciate the experts today that are volunteering their time and the patient for taking the time to share their story. So thank you from all of us. What I'll do is just spend a few minutes walking through the work of the ABTA that we do in concert and in huge collaboration with so many. First, for those of you that are newer, this is the mission of the ABTA. Our vision is a world where no life is lost from a brain tumor. That's our vision. And how do we achieve it? Or what is our key core approach is to improve, to extend, and to ultimately save the lives of everyone diagnosed with a brain tumor. And what I'll do today is talk a little bit about how we do it. First, research. Second, patient services. So let's talk a little bit about our research program. It is our legacy. We were founded almost 50 years ago in March of next year. So we have a investigator-initiated peer review process where we fund all tumor types and all ages, and that's including metastatic disease research. And you can see on the screen the dollar, the projects, and just look at the number of researchers we have funded and whose lives we have heard so many times tell us how a grant from the ABTA early in their career had led to the robust lab that many of them have today. So again, we fund all ages, all tumor types. Next, let me just share, since we do have just a brief time, one of the true gems of the ABTA, and that is the ABTA's Alumni Research Network. So you see on the screen the last time we were able to get together in person back in 2019. The good news is we are meeting in September in person again in Chicago, 2.5 days, where we bring together investigators we have funded at one point in their career. So these are early stage investigators generally, but then there's a number of other more senior investigators where they network, they learn, they sometimes collaborate on grants and other papers even. There are many examples of that, some of which there are listed there on your screen. So we're proud that we don't just fund investigators. This is one way we have heard from them. It's a really important 2.5-day session. And of course, they stay in touch throughout the year. And next, let me tell you a little bit about the ABTA's patient services, the support that we give to patients and caregivers, of course, free of charge. And it's all because of the generous support from people all over the country. And there are so many services and programs, the quality, the scope. I'm really proud of them. Everything can be found on abta.org. It's our website. Let's start with something newer, our Brain Tumor Treatment Center Guide. You can find on the webpage information about brain tumor centers all over the US. And these are only US centers at this point. It is self-reported data, again, available on our website and available to everyone free of charge. We think it's a valuable tool, and I commend our team for this work. And it's an ongoing commitment to stay in touch, to make sure it's current. You can see at the bottom of the slide on the screen some of the data that you can find for brain tumor centers all over the US. Next, we also provide education materials, brochures. In addition to being in English, we also offer most of them in Spanish, but not all. And as revenue allows, we want to make more of our material available in more languages. It's just really important for us to make that available. They're free to patients and families. They're also available for you to download on our website, abta.org. And as I mentioned, we're going to translate more of our material in more languages. Next, an example of one-on-one care that we provide is our care line. We have trained staff that will talk to you by phone, by email, by text messaging, to listen, to provide some emotional support at a critical time that you may need. If we can't provide the service, we can refer you to services such as financial assistance, and other examples of a pretty strong referral network. We also get calls from healthcare providers and other professionals. We are not here for medical advice, but we are here to support you, to inform you, and to give you the information you need to have a better dialogue with your providers. And we're based in Chicago, and you see on the screen the toll-free number, and again, the email that we also can provide that one-on-one conversation with you and have ongoing dialogue, not just a one-time conversation. Next, one of the many ways that we reach larger audiences is through our online support community called The Connections. And you can see on the screen, it's growing, and 34,000 members now. A big chunk of them are very active. We are told by the folks that host this site that our community is one of, if not the most active on this online support community. So you're welcome to participate, to just get online. You can also join anonymously if that is something you would prefer to do. Of course, it's 24/7, and it is also free of charge. Next, throughout the year, and generally they're held monthly, we have free webinars that are just they're in response to requests from brain tumor patients and caregivers. What are the kinds of things that you want to hear from us? And then our team, Mindy and Emily and Nicole and others, they go out and they recruit the leading experts in the country, and they generously volunteer their time to share information. And again, they're free, they're monthly. You see on the screen an example of what James offered to us at the beginning of the year, coming up, in July. We've had these throughout, all the way back to the beginning of the year. They're all posted on our Anytime learning channel on YouTube. Go to, as you see on the screen, where you can find them, but abta.org is a great place. Next slide, please. We have these patient and family meetings. This is the third of the year and our final one. As I said, we can't thank Mike and the other doctors that are available today that have volunteered their time to get today. It's very generous of them. Our team puts a lot of work into promoting these very important half-day sessions. So as you see on the screen is a quick summary. Next year, Emily was referring to how much we miss not being able to spend time in person with people. So next year, we're planning on 3 patient and family meetings, and we're planning at this point and having them in person in various parts of the country, but also available online. So, but we know many people will not be able to, attend the New York or Tampa or LA in-person meetings, but they'll be available online next year. Stay tuned for announcements about those. Next, the largest of its type national conference is the ABTA's national conference. And unfortunately, with the pandemic, this too has been virtual, but we've had record registration, record interest. The people that participate in our national conference in the surveys have told us how meaningful and important the content has been. If you haven't yet done so, our 2022 National Conference is September 9th and 10th, online, free of charge. Please sign up today, abta.org. You will find the link to sign up if you haven't already. You can look on the screen at these 2 amazing leaders in the brain tumor community that have agreed, that are serving and working closely with Emily and Mindy and Nicole to develop and to finalize everything for the conference. It's going to be exciting. We're looking forward to it. We are planning on having our 2023 National Conference in Chicago in person, but also available online. So again, if you haven't signed up for the National Conference, please do so today or call us if you need more information. Next, we also recruit, train mentors. So you see on the screen how many trained mentors we have. And they're not just brain tumor patients, they're also caregivers to brain tumor patients. So we'll match you and then you get together virtually as often as the both of you would prefer. So if you want to serve as a mentor, you can contact us. If you need a mentor, please contact us. You can see how many matches we have made, and we are really pleased to be able to offer this. So call us, email us, and let us know if you want to serve as a mentor or get matched with a mentor. So we cannot do the research and the patient services that you see, and you've heard a little bit about. I know it's very quick. Without the involvement of so many dedicated volunteers and a few really wonderful and caring ABTA staff. So let me just tell you briefly how you can get involved if you wish. First, we have what is called Team ABTA. And that is, we will support you if you want to run in the New York Marathon or the Chicago Marathon or other kinds of endurance events, and you would raise money on behalf of the ABTA, and we would organize your efforts. We also, for any of you anywhere, whatever your interest might be, from bowling to golf to tennis, you can put on your own fundraiser, raise the money for the ABTA, and we can provide some level of support to anyone that wants to raise money for the mission in your community, with your family, your friends, your colleagues, any size, any scope, anywhere. We will do all we can to support you. Again, call us, email us, and we'll give you more information. Next, our signature walks and runs are called BT5Ks. This year we have 8. We have New York, Twin Cities, LA, and San Francisco remaining. We would welcome more volunteers in those markets. We also have a virtual BT 5K, so of course anyone can sign up for that. Where can you find it? abta.org, and you can get more information about each of those events. I just want to thank those of you that have supported the events that have already happened. That is fabulous. And in fact, Mike Vogelbaum and the Moffitt folks had the largest team at our Tampa BT 5K. So I want to shout out to Mike. Next, we have other volunteer opportunities. As I mentioned, you can serve on a BT 5K. We have the mentor program. We have outreach specialists. We have one volunteer manager. So Sherry, you just contact any of us at the ABTA. Sherry will follow up with you and share in a bit more detail the many volunteer opportunities that we have. And some are in person, some are virtual. We will match how much time you have, whatever level of expertise you may have. You certainly may not be a neurosurgeon, but you may have a skill as a photographer, and you're willing to take a few hours to go and take some photographs of an event or of an activity for the ABTA. But there are many ways that you can volunteer and help the ABTA as a volunteer in raising money, and you might have a unique skill set that you can help. And so many of you are probably very good at social media, and boy, do we need to get you to promote the free services and opportunities of the ABTA. So that's just a quick overview, and I know I moved through it pretty rapidly, but abta.org is the place. Next is a place for you to stay connected with us. It's a free e-newsletter called Mind Matters, and we would welcome anyone to sign up free of charge to get this virtual newsletter to your account, to stay in touch with the work of the ABTA and the really key events happening in the brain tumor community. We ask you to follow us on Facebook, on Twitter, Instagram, LinkedIn, whatever you are on. We would welcome you to help promote the work of the ABTA. And then one obvious way next that you can help us is to donate anything from a small amount to a large amount, to introducing us to individuals, to foundations who might have an interest in supporting the work of the American Brain Tumor Association. Myself or Nicole or other people on our team will work with you. All you have to do is make the introduction. We will take it from there. Thank you. Thank you for your time. Thank you for being here. And we all look forward to a great day and to learn much. Thank you, Emily.
Thank you so much, Ralph, for sharing with us all about the American Brain Tumor Association. I'm going to now hand it over back to our chair, Dr. Michael Vogelbaum, to introduce our next session and our speakers.
Well, thank you, Emily. And before I get started, I just want to thank Ralph again for his excellent leadership of the ABTA. Ralph has been a fantastic CEO, very energetic, as you can see, has really been helping to drive the mission. What most people don't know about Ralph is he's also secretly a top-ranked tennis player. And not sure why the US Open has missed out on him, but they really should think twice and send him an invitation. He's really good. All right, so we're going to start with our first session, this titled Laboratory and Translational Research. And we have two speakers who will discuss what constitutes laboratory and translational research, why it's important for patients with brain tumors, how it's done, and paid for. We have two speakers today, Dr. Justin Lafia, who leads a translational cancer stem cell research laboratory and is professor of the Lerner Research Institute, professor in the Lerner Research Institute and scientific director of the Rosella Burkhart Brain Tumor and Neuro-Oncology Center at the Cleveland Clinic. Dr. Lafia and I have been long-time research collaborators. And as part of that, we often travel to the same national and international scientific meetings. And one of my favorite travel experiences with Dr. Lafia was when we both descended into an Icelandic volcano during a 36-hour layover while returning from a brain tumor meeting in Norway. Joining Dr. Lathia is Dr. Craig Werbinski, who's a tenured professor at Northwestern University and chief of neuropathology. He has an NCI-funded brain tumor bank that has supported over 120 intramural and extramural research projects. Dr. Werbinski is originally from Buffalo, New York, and he's well known in the brain tumor field to be the foremost connoisseur of buffalo wings. Please welcome our speakers. Dr. Lathia will present first, followed by Dr. Werbinski.
Okay, good morning, everyone. It's a real honor and privilege to spend some time with you this morning. Wanted to sort of go over this idea of what laboratory research entails, and really the process.
We're giving you remote control now. Click the slide 2 times.
Okay,
There you go.
so again, nothing to disclose here. What I'm hoping to cover this morning is the idea about what is laboratory and translational research and what does it mean? I also want to touch on why it's important for understanding the disease, but also thinking about how we can develop better therapies. I want to tell you a little bit about how it's done, and then I think it's important to really talk about how it's funded. So what is laboratory research? So it's generally focused on a specific question. So I gave you an example here. Why do brain tumor cells grow so fast? Why are they resistant to things like radiation? And these are usually hypothesis driven. And by that, what happens is the scientific team has an idea and they really design experiments to test that idea. So that's really the fundamental basis of laboratory research. And again, as you can see in the next bullet point, the experiments are designed to answer the question, and I'll go through that a little bit more in a second. It's really done by using laboratory models, and these vary. It can be things like genetic information from a cohort of patients. It can be using cells in a culture dish. So there's a wide variety of approaches that a scientific team would use to do the experiments to test the hypothesis. Right, and again, giving you some examples below. And then really what happens is when there's enough scientific information or data generated, the team usually comes to a conclusion and there's an opportunity to reevaluate the hypothesis. The truth is, the exciting part about science is we often come up with theories or experiments that actually aren't correct. And by learning from these failures, it really helps us do better science and do better experiments. So what is translational research? So this is a term many of you may have been exposed to or heard. And really the idea is it's about not just generating knowledge, but actually trying to figure out a way how this can be translated into a clinical application. And this can happen in a variety of ways. I've given you one example here of actually using human samples. So, you know, if a tumor is removed, there can be some genetic assessment that would actually count as research involving human samples. So here's a schematic in the bottom left, and I took this from the Breast International group. But the idea is you can see basic research is on the left and clinical research is on the right. And in the middle lies this idea of translational research. So that's the idea. And again, it's a term that's used a lot, but going bench to bedside. And it gets a little bit more complicated. So if you look in the bottom right, this is an image from NCATS, which is an affiliation with the National Institutes of Health. And it's this idea of this continuum from starting with basic science research and really ending up into a patient, into clinical trials, which I suspect you're going to hear more about later today. So that's translational research. So why is it important? It's important for a lot of reasons. I think the first is to understand the complexity of the diseases we're working with. Brain tumors are very complex for a variety of reasons. And I think that this sort of research is providing insight into that. The second thing is it really helps us develop more effective therapies, right? To create these therapies, we need to be in a position where we better understand the disease. And really that's sort of captured here is that my argument is a lot of the new therapies are based on observations that have come from the lab, have come from very focused hypothesis-driven questions. So how's it done? I want to give you a real-world example of some of the things that my team is thinking about. And I should note that actually these studies were initially supported by the American Brain Tumor Association. So the question we had years ago is why is there a difference between male and female glioblastoma patients? There was a lot of observations epidemiologically, but we wanted to get a better understanding. So what I'm showing you here on the right are incidence curves of patients with all gliomas, and then on the right is glioblastoma. And there's two groups, right? There's a red group and a blue group. And it turns out that the difference has nothing to do with the genetics. There's no genetic mutation that's different. It has nothing to do with environmental factors. What it actually comes down to is biological sex. So if you're a male or a female, there seems to be a difference. And this really is shown better if you think about response to therapies and sort of overall trajectory. So this is what was standing in front of us, is what's the difference between these patients. So one of the things we thought about in a hypothesis perspective is we thought the immune response would actually be different between males and females. And what you're seeing in the center is a table taken from a review that really made the argument, if you look at males and females, females actually have more autoimmune diseases, and you can see some of them listed on the left, and males have a little bit more of a bias towards non-reproductive cancers. So really the question was, what is the difference between males and females? And the hypothesis was the immune response will be different. So we went ahead and tested that rigorously, and the truth is it took us about 4 years to come up with these conclusions through a lot of testing. So what you're seeing on the left is a summary of what we found in mice that were used as brain tumor models. And what we actually found was that males had a different immune response in their tumor compared to females that had a different immune response peripherally. And we were really excited by these findings. We were able to actually test different sex-specific drug regimens. But what was also satisfying is if you look on the right-hand side, we were actually able to show using patient samples. So these are patient samples we got from our neurosurgery team. We got pieces of tumor, we got blood samples, and we're able to show that what we found in mice actually repeated with what we found in humans. And we're currently using this information to develop some really early phase clinical trials to try to leverage what we learned here, really to translate what we learned. So I want to really end with this idea of how is it funded? Laboratory research isn't cheap. Someone who runs a lab, I'm responsible for paying the salaries of everyone in the lab, providing all of the reagents they need. And this is actually funded mainly through the US government, through the National Institutes of Health, as well as the Department of Defense, as well as the National Science Foundation. And again, there's 27 NIH institutes, and several of them fund brain tumors, such as the National Cancer Institute and the National Institutes on Neurological Disease and Stroke. But it's also funded through private foundations. So a lot of these government grants are quite difficult to get. The success rate is, on average, somewhere between 10 and 15%, depending on the institute. But private foundations like the ABTA can really step in. And I can speak from personal experience. When I was still training, I actually got a fellowship from the ABTA. That was my first grant that really helped me stay in the field. And again, I just told you about these differences between males and females. That initial idea was actually supported by the ABTA to really provide a catalyst for future work. Now, there is also institutional support and private donations, but again, these vary from institute to institute. And in general, this is really the funding landscape. And I will say that the other thing to remember about funding, it's not about just funding a lab and providing the resources, but really a lot of the next generation of physicians and scientists are being trained in these labs. So one of the things that that we also do is we view our lab as sort of creating the next generation of scientific researchers and importantly, brain tumor researchers. So funding is really important here. So again, this is the disclaimer. I'm happy to stop here and turn it over to Dr. Horbinski, and I'll hang on later for some additional questions.
All right, Dr. Herbinski, we're going to give you remote control.
Great. Well, thanks everybody for attending and for giving me the opportunity to share with you a little bit about how brain tumor banks actually support the kind of research that Dr. Lathia just talked about. For example, at Northwestern, we have a nervous system tumor bank that's relatively young compared to a lot of other programs, but we made up for it in activity because since 2014, we've collected over 2,500 tumors as well as matched blood, and that actually includes both pediatric and adult tumors. And we're closing in on 3,000 tumors, actually, getting very close. And we've used all of that material to support 126 and counting active and completed research projects from nearly 50 investigators, including extramural investigators like Dr. Lathia. Now, here's how it works. So a potential candidate for the biobank, meaning somebody who comes in, presents with symptoms, that radiologically they look like they may have some kind of a tumor. Our staff will obtain consent from them. And if they don't want to consent, that's perfectly fine, then they don't have to. But if they do consent, then the next step is right before when they're in the operating room, the anesthesiologist will do a pre-surgical blood draw. That is then our staff takes and separates it into various components. And that's exactly what these little vacutainers, and we store them and freeze some others we give freshly to other researchers who have IRB approved protocols to study things like immuno-oncology, that sort of thing. And then once that's done, the surgeon will go in, and the neurosurgeon, of course, will be doing the tumor resection. Once the tissue biopsy is given to the neuropathologist, sometimes that's me because I'll often be on service, that will render an intraoperative diagnosis and will determine whether or not indeed this is actually a useful piece of tissue. And if it's what we think it is, if it's a tumor, then we'll collect it, we'll grossly evaluate it and partition it onto all these different things. So portions of it will be frozen into little aliquots. Other portions will be dispensed freshly to researchers if they need fresh tissue for their own laboratory research. We'll develop sometimes patient-derived xenografts, so we'll take the tumor and grow it in mice so that we can propagate it and then use that tumor tissue and test new drugs and new therapies on it. Sometimes we'll develop cancers. Sorry, my apologies. Sometimes we'll develop cancer stem cells or informally fixed paraffin metatissue sections. And that's where we'll take so a lot of this is done by that's Katie McCourtney. She's our biobank operations manager, and she's putting frozen tissue into liquid nitrogen. And then we'll also do tissue blocks. So we'll take tissue blocks just like clinical blocks, and that's what they look like. We have a separate swipe card protected room in our bank that stores all of these tissues, and you can see the tissue cassette right there. And that's the histology that will produce and study the tissue to make sure that it is what we think it is, that the tissue quality is good. And on top of that, on the clinical setting, we'll do a battery of detailed molecular tests to confirm the diagnosis, to provide prognostic information. And all of those molecular tests are annotated in biospecimens in our database. So for example, some of the annotation that we do involves next generation sequencing, genomic copy number profiling, and genomic DNA methylation profiling. Those are the big three that we do at Northwestern. So for example, the next generation sequencing panel that we use it's not sequencing the entire genome of the tumor. It's focusing on hotspots, high yield spots that we know are frequently mutated or altered in some fashion in various kinds of tumors. And in the case of brain tumors, you can actually diagnose a lot of these tumors just by the patterns of genetic alterations that they have. This can include various kinds of mutations, what we call single nucleotide variants, or various extra insertions or deletions of DNA across the entire genome. Some parts get copied way too many times, so instead of having 2 copies of a gene, you might have 50 or 100 copies. That would be an amplification. All sorts of really bizarre things can happen in the genome that drives cancer and makes cancers more malignant. So it is indeed, cancer is indeed a genetic disorder. That's what it ultimately is. Now, more recently, we interrogate things like microsatellite instability and tumor mutation burden, because there is some evidence that, at least in some patients, this is really important for predicting their response to immunotherapy. Because if they have a hypermutated tumor, a tumor with lots and lots of mutations, they may be more sensitive to immunotherapy. So here's just an example of what something might look like in terms of copy number profiles. We all have 26 chromosomes, and including the X and Y chromosomes 1 through 22, and 24. Anyway, we have 2 copies of each chromosome, chromosomes 1 through 22, and then 1 copy of X, or 2 copies of X and 1 copy of Y. And what's interesting, and this is fascinating in its own right, is that certain copy number alterations will be consistently recapitulated across multiple certain kinds of brain tumors. Like for this one in particular, I can look at this copy number profile and determine right off the bat that it is a glioblastoma, because it's got a gain of this little segment of chromosome 7 right here. That's the orange arrow, especially amplification of EGFR right here. And then we've got deletion of CDKN2A and B right here, which is a very powerful tumor suppressor, so it's lost, as well as loss of chromosome 10. So that combination right there is a glioblastoma 10 times out of 10. In contrast, here's what an oligodendroglioma looks like. You've got a loss of chromosome 1p and loss of chromosome 19q. There's also a little bit of loss of chromosome 6 in this particular case, but really it's that combination of 1p and 19q loss that diagnoses the tumor as an oligodendroglioma, and it really doesn't have any of those other alterations that we saw in the glioblastoma. And we use all of that data. We store all of this data and annotate each one of our cases. Each one of our banked cases has this, so that when researchers come back and say, hey, I don't need just to study glioblastomas, I need to study glioblastomas that have EGFR amplification. Well, then we were able to give them those exact kinds of tumors, exactly what they need to do their work, like this case. And another huge breakthrough that warrants specific attention is the idea of methylation-based classification of CNS tumors. Now, what does that mean? Well, methylation is a chemical modification that can happen to DNA. It's just a little carbons are attached to millions of different sites across a cell's entire genome. All of our cells have this. This is perfectly normal. And it allows cells to decide what kinds of genes are going to be turned on and what are going to be turned off. And so that you get these little chemical modifications, these little methylations can happen all over the place. And what's interesting is that at any given site, it's either methylated or it's not, and it really doesn't tell you anything informative about just one little site. But when you can screen hundreds of thousands of these sites all at once, which is what we can do in these arrays, all of a sudden a pattern emerges, forest for trees pattern emerges that reveals the identity of that tissue, that reveals the identity of that tumor, and allows us to prognostically stratify different kinds of tumors that otherwise look almost identical under the microscope. And that's exactly what was done with this classifier development back in 2017 that we still use to this day. And that's exactly what it is. They bioinformatically, using very sophisticated analyses, figured out what patterns of methylation are associated with certain kinds of tumors and certain more or less aggressive tumors. And this is what it looks like when so I sign out these molecular reports all the time. Every week I sign out at least one batch where we've got this two-dimensional plot, and you see all these little clusters of colors and they're little dots. Each little dot represents a tumor in the library, in our methylation classifier library. And each of these different clusters is a unique, different kind of tumor. So for example, down in the lower right here, we've got all these different yellow or gold colored. Those are all the IDH mutant gliomas. In the center or off center, you see the green, the larger green cluster. That's 6 or 7 different types of IDH wild type glioblastomas. And then we've got way up here at the top, a couple of different clusters of medulloblastomas, and down over here, there's a couple more clusters of medulloblastomas. The red clusters that you see scattered all over the place, those are all kinds of different ependymomas, and there's all sorts of meningiomas are in here too. So what's interesting, you can take a new tumor, an unknown tumor, run methylation classifiers, and see where that tumor lands in this whole scheme. And that tells you the identity of the tumor, even if we can't tell otherwise under the microscope what it actually is. And all that data we keep and store and use on a research basis, just like Dr. Lathia said, so that we can study these and understand these tumors a lot better. One example in particular that I'll show a brief example on how biobanks can affect brain tumor research and our understanding of brain tumors is a study that I started back in 2015 when I became the biobank director at Northwestern. And the idea here was to take patients courageously donating their brains and spinal cords at the end of life, and we would collect the tissue, we would study the pattern of tumor spread, and with the idea that we're going to study mechanisms of therapy resistance. You know, why does the tumor develop resistance to radiation and chemotherapy, that sort of thing. So one of the things we found that was really surprising was, even though the radiology made it look like the tumor more or less stays near the original site, what we found under the microscope is that the tumor is spreading literally everywhere in the brain, kind of like ants crawling through a computer. And we were able to show that even though in the old days, in these old studies from 83, 1989, and 1991, most patients were actually dying of the tumor getting big and squeezing the rest of their brain. Now that's not the case anymore. Not so many patients these days are dying of mass effect from the tumor because we're getting very good. People like Dr. Vogelbaum are very good at controlling that. But what they're dying of instead is tumors spreading to critical sites like the brainstem, which controls your speaking, it controls your swallowing, it controls your heart rate, your sleep-wake cycles, and all that. And what we found was that actually, whereas heavy brainstem invasion used to not be a thing, now that in our cohort, that's quite common. And it's not, you can't always see it radiologically because they're individual tumor cells just crawling through, and the only way you can see it is under the microscope. So what we're seeing here is that no matter where that tumor arises, it's got at least one direct route of access to the brainstem from anywhere in the brain. And that is a big contributor to why people are not responding to therapy. Because if you got your therapy and it's focused on the original tumor site, well, if it's not addressing this phenomenon of tumor spread to a vital structure like the brainstem, that's going to be a problem. And this is really salient because when we take those patient-derived xenograft models, like I mentioned before, we take the tumor and we put it in the mouse brain, and we grow the tumors because we're trying to test a response to therapy. A lot of these tumors, this is a cross-section of a mouse brain, and this is a human tumor growing in that mouse brain. It doesn't spread in a mouse brain like it did in the human brain. It grows as more or less a solid ball or well-demarcated mass, and it does squeeze the brain. So mice, when we were treating these animal models and we're trying to determine a therapy effect and everything, those models are killing by mass effect. They're not killing by brainstem extension. So it's the kind of thing that we have to keep in mind now. That, you know, every model is interesting, but no model is perfect. No model is 100% accurate. And when we're trying to come up with new therapies and trying to figure out why drugs that worked great in mice fail clinical trials, well, this could be a big reason why, because it's not addressing that diffuse spread. So at any rate, that's just one example of how biobanks can support research and even start new research ideas. And I'll take any questions.
Great, thank you so much, Dr. Hrabinski and Dr. Lathia. We will now proceed with our Q&A. If you have a question, please type in your questions by selecting the Q&A button on the bottom of your Zoom control panel, and Breen will be filtering through the questions. Thank you so much.
So
Great.
while
Thank
we're
you
waiting
both.
for, sorry, go ahead, Ambry. Do you have a question ready?
Yeah, well, I just want to thank both of you for a wonderful presentation. I think it's really exciting to see how much can be done in research. But when we look at brain tumors versus other tumor types and other cancer types, it often seems like advancements in the brain tumor space happen much more slowly. What do you think is the biggest barrier in advancing treatment options, trials, and research for brain tumors?
If you don't mind, let me take that first from a basic science perspective. These tumors are growing in very eloquent areas such as the brain. And one of the things we didn't really talk about is there's a barrier, there's a protective layer on the outside of the brain called the blood-brain barrier, right? This actually keeps a lot of things that we don't want in the brain out of the brain. But this also presents a challenge when we're developing therapies. We actually now have sort of an extra level that we've got to clear. Not only do we have to show that these therapies, these compounds, these strategies are effective against the tumors, we got to get these across the blood-brain barrier. And I think that's a major challenge. So if I were to point to one thing, I think that's what I would point to.
I would echo that. That's certainly a serious problem. I would also say, like for example, what I mentioned with the research that we were doing on postmortem brains in glioblastomas is just this idea that the single cell infiltration, the way that these tumors see other kinds of cancer, are what they do is they spread en masse. They spread in clusters of cells. They spread as cohesive sheets and masses of tumor invading local structures in other organs like the colon and pancreas and everything. Gliomas are unique. They're the only solid tumor that actually spreads on an individual cell basis. In other words, you can look at a field, a microscopic field, and 90% of the cells could be normal cells that belong there, like glioma, or just neurons, glial cells, endothelial cells. But like one little cell over here is a tumor cell, another over here is a tumor cell, another here. And when we study them in the research setting, we see that they're crawling along this little like amoeboid movement. So it really is. When I talk to patients and the next of kin, sometimes with this research program that we're doing, I describe it as being like ants crawling through a computer. So from a distance, the CPU might look fine, the power looks fine, there's nothing overtly wrong with it, but it won't boot up, it won't start up properly, and it keeps crashing. And so it's only when you take that computer apart, you look at the motherboard, and you realize under the microscope, oh, there's little bugs crawling all over it. No wonder it doesn't work. That's what we're seeing, and that's a sort of tumor spread that you can't see radiologically, certainly not to its fullest extent. And that is a unique pattern of behavior in gliomas in particular relative to other kinds of tumors. And I think that is a big challenge that the field has, is trying, how do you address that?
And a follow-up question to that, Dr. Herbinski, do you find that that's similar for metastatic brain tumors as well, the difference that you see in how glioma spread and grow?
Yeah, metastatic brain tumor doesn't spread like gliomas. It's little pockets or little clusters or spheres of tumor cells that more or less stay in and around the blood vessels. So even the blood-brain barrier isn't so much of a problem when you're trying to treat metastatic cancer. It's just that the cancer is resistant to everything you try. Because in those little spots where the tumor is residing, the blood-brain barrier is broken down. That's how it got into the brain in the first place. It broke down the blood-brain barrier in those little spots. So that's not as much of a concern really when you're trying to treat mets. It's just that metastatic cancer is really ultra tough cancer that resists everything that we give it.
Thank you so much for that. And Dr. Herbinski, you talked about donating tissue to a biobank like the one that you manage. Where can patients find information on where they can donate posthumously their brain or their body for research?
If anybody who has gotten any care, for example, at Northwestern, it would be eligible for this program at Northwestern. And the hospital will cover the cost. That's just part of the hospital's mission. But there are a couple of other tumor banks that do have postmortem collections. Like I know Duke does this, UCSF does this in San Francisco. Most don't, though. Most brain tumor programs don't invest this kind of the kind of resources that are necessary to do this sort of thing. And I have had people from, very courageously from other institutions or even all across the country ask, how do we get involved in this? Can we send our brain spinal cord, to you? I know it's a very awkward conversation sometimes, but these people I find are much tougher and more willing to engage in this than sometimes even we are as clinicians, but it is a challenge because as far as billing is concerned, if the patient didn't get any of their care at Northwestern, then the autopsy costs several $1,000s, and insurance, of course, is not going to cover that. So there are some barriers.
So is there a centralized place where tissue can be sent to? We have, I believe, a patient or an attendee here asking how their donation can be found. They had filled out the form to take part in a donation, and they're wondering if there is a database and how are you locating that tissue and finding it for your research?
So you're talking about like tumor tissue that's donated? Well, our bank, we have a very detailed annotated set of going on 3,000 specimens now. So if somebody anybody can ask us. We have an online request form if somebody wants tissue from us, even from an external site. They have to provide all the IRB documentation to prove that the research has been vetted by an institutional review board, for patient safety and privacy and all that. There is no central repository for brain tumors. Now, we are part of some consortia where, like AlloGo-Nation and Children's Oncology Group, for the pediatric stuff, where if somebody wants a really rare tumor tissue, they can go to that central site and say, okay, here's a request, here's all the participating banks, who has that tissue, who has something that they can spare and they can share. So we do participate in that, but there's no central, nationalized repository.
Thank you so much for providing that understanding of how that works and the collaboration that takes place. So Dr. Lathia, you talked a lot about the translational research and how that works. What role can patients play in supporting translational research?
I think most places will have an IRB when the patient's treated, which asks for, were they willing to donate their tissue? Are they willing to donate blood? I think that is really the first step. If patients are willing to do that, what we can do is we use that information to actually generate new hypotheses, which we then go back into the lab and test. And then we really like to validate our hypotheses in that way. So I think, to me, the first step is, when they're being treated, to be willing to provide some, blood or tissue for research purposes.
And if I could add to that, you're going to be hearing a little bit later from folks who work on the other side of that equation as well, and that is going from the clinic to the laboratory. So participation in certain types of clinical trials can provide a lot of tumor samples, blood samples that are critical for translational research. So it doesn't just go from the lab to the clinic, but it also goes from the clinic back to the lab. And you'll hear about that in the next session.
Thank you, Dr. Vogelbaum, for adding that different perspective. Another question we have here is asking about the tissue that's donated. How long is that tissue stored for, and how long can it be used?
We store tissue indefinitely. There is no limit to how long we'll store the tissue. Now how useful it is depends entirely on how well it's been stored. So, for example, when we store things in the vapor phase of liquid nitrogen, like I showed in the early image of one of our biobank technicians pulling something out of a liquid nitrogen tank, that's like a minus 160 degrees Celsius. So pretty much all molecular activity stops, nearly all activity stops, so there's no decay of tissue. You can take something that's 20, 30, 40 years old from a freezer like that, so long as it's maintained in that liquid nitrogen state, and you'll be able to do all kinds of DNA and protein analyses and everything. It'll still be good tissue. Paraffin embedded tissue blocks, like when we look at tissue slides, we have that little stuff that's embedded in paraffin wax. That doesn't last as long. In our experience, you really start to lose a lot after 10 years. So beyond 10 years, trying to do any kind of DNA analysis or anything like that is very sketchy, and you're liable to not get really good results. But really, as long as it's been frozen properly, the biggest problem isn't even once it's frozen. Once it's frozen, it's set. It's getting that tissue frozen quickly once it comes out of the operating room. And that's where you have to have a really robust pipeline, and good infrastructure, good communication between the OR staff and our biobank staff. And they have to really be committed and serious about getting us that tissue, because the data shows if it's not frozen within 15 to 20 minutes after being removed from the patient, you lose a lot of tissue quality.
Thank you so much. And on that same note, I believe on one of your slides, Dr. Herbinski, you mentioned finding a good candidate for the biobank. What qualifies a patient's tissue or a patient as being a good candidate for a biobank?
It's the preoperative phase where patient presents with certain symptoms. You're usually going to get some kind of radiology scan, CT or MRI, and you see, oh, there's something in the brain or spinal cord or what have you. And we think it's this kind of tumor based on its radiologic appearance or that or the other thing. And so then we'll consent the patient before surgery. So we don't know 100% sure. We're not 100% sure what you have. That's what the surgery is for. That's what neuropathology is for. But whatever you have, would you be willing to donate leftover tissue, whatever's not needed for the actual diagnosis? Would you be willing to donate that tissue to us for future research? And we are very specific about what kind of research is permitted with that tissue, what's not. So we're not, nobody's identity is going to get divulged or violated in any regard. And like, for example, sometimes once in a great while, we'll have a tumor, we thought it was a glioblastoma, and it ended up being a massive stroke or something, or a viral disease or multiple myeloma or sclerosis or something like that. We just had a toxoplasma case this past week that looked like a GBM. Well, those cases we're not going to be able to really use for research because, it's an infectious disease, and we're a brain tumor program. So we wouldn't bank something like that. We would just say, okay, now that we know what it is, we're going to skip this one. But that doesn't happen too often. I say way over 90% of the time, it ends up being some kind of tumor. And we take all comers. So we take everything from metastatic cancer to gliomas to benign tumors, meningiomas, even pituitary adenomas. We collect everything because we don't know, someday, 5-10 years from now, somebody might have a project that needs, 500 pituitary adenomas, for example. Well, you can't start collecting at that time. It'll take you forever to get that. You got to start ahead of time and guess that maybe someday somebody's going to want to use this tissue.
If I could add just real quickly, one of the points that I think has been implied but not said explicitly is that not every hospital has a brain tumor bank, and in fact, these are quite specialized programs that are present only, for the most part, only in the larger specialized brain tumor centers, and then not even all of them have those. So hopefully you all understand how important these are, how valuable they are in terms of getting to where we need to get, which is to develop cures for brain tumors. But the only way that we can populate these banks is by collecting at these very specialized centers that do this kind of research and bank the tissue that's necessary to do the research.
So Dr. Vogelbaum, a follow-up question to that. For patients who are not at these larger institutions, what can they do? How can they support biobanks and the research that's being done?
I think that's a great question. There are many levels to that answer. Sometimes it means that they do need to have some of their treatment done at one of those centers, particularly for some clinical trials and the most cutting edge clinical trials, the ones that are developing new therapies. They're really only being done at those major centers, and then those centers will provide opportunities for tissue or blood or other sample collection as part of those trials and as part of the care and the like. That's probably the easiest way. I'm not sure, as Dr. Herbinski said, that when you're having a surgery done at a community hospital, no matter how good of a community hospital, they're not going to be set up to collect the tissue quite in the same way to be collected under the conditions that will allow for advanced research to be done on the tissue. Most of it is put into what we call paraffin embedded blocks. It's fixed. A fixative is applied. It's put into a block. There is some type of research that can be done, and maybe you guys can comment on some of the larger genomic types of studies that can be done using those blocks. But the more detailed studies in terms of tumor function, immunobiology, much harder to do with that kind of tissue.
Dr. Herbinsky or Dr. Lathia, did you want to?
It is. So that's the importance of having really detailed and rigorous and pretty much comprehensive molecular workup like what we do. So we got all the mutations, we have all the copy number alterations, we have all the methylation profiling now that we're doing. And because of that, and we store all those tissues, and we also track all the patients. We see how they do. How did they respond to therapy? You know, how long did they survive? I mean, we've got patients who we thought had one kind of tumor, like 5-10 years ago, and they're still here, and we're like, well, we're glad you're here, but we have to be honest. We thought you would have, with what your original diagnosis was, we thought your outcome would have been a lot worse. So then we go back to the original tissue. We probably do this a couple times a month now because we have all these new molecular tests that we didn't have before. And we'll do, like kind of in hindsight 2020 molecular testing. You're like, oh, your tumor wasn't nearly as aggressive as we thought it was. Histologically it looked really bad, but by molecular, it's very treatable. We now know this particular subtype responds to a certain kind of therapy, radiation, what have you, or it just doesn't recur easily. I mean, just for example, we released a new 2021 WHO classification scheme. I helped write one of the chapters. In the prior scheme from 2016, we had about 85 entities that could arise in the brain and spinal cord, 85 different kinds of tumors. A lot of them are very rare. We're up to 127 kinds of tumors in the brain and spinal cord. It's not that those tumors didn't exist before. We just weren't aware that they were unique entities until we did all this detailed molecular analysis and said, you know what? This tumor might look bad, but it's got a very favorable molecular profile, and that greatly changes treatment. So that happens all the time now. All of our cases, we're really getting a lot better at predicting with much more precision how a patient's actually going to do.
And one of the best examples of that, where people in the community were able to participate, were the trials done in the 1990s on what were called anaplastic gliomas at that time, grade 3 gliomas that were just all lumped together. And it was comparing one treatment to another. But then because the tissue blocks had been collected, even though they were just paraffin embedded fixed tissue, they were collected and analyzed centrally as part of that study. It was discovered that there was this marker that could identify one group from another. That's the 1p19q story that Dr. Herbinsky referred to earlier, where he showed an example of an oligodendric glioma. That was revolutionary, because now we had the ability not only to sort out some of the better acting tumors from the bad actors, it actually altered the treatment as well for those tumors. So it's led to many more clinical trials since then. So sometimes it can work where the community and specialized centers can come together and really provide material to make a great advance. Most often it's going to happen in the specialized centers though.
Dr. Lathia, I saw you came off mute. Did you want to add anything?
Yeah, I mean, in terms of what we learn in the lab, I think having the infrastructure is really important. It's really difficult to make new models based on patient tissue, to do the high resolution immune analysis, unless we can get samples very quickly. So I think for us, the infrastructure, as Dr. Herbinsky alluded to, is really empowering a lot of what we do.
All right, I think we have time for one last question. We have a question here asking about stem cells. Can stem cells that were frozen previously be used for research?
Yes, we do that all the time. We take tumors and develop stem cell cultures from them and then freeze them. And sometimes we'll implant them into mice to try to grow solid tumors that we can then treat with chemotherapy and radiation. or we'll hand it off to, stem cell researchers like Justin Lothier or some of my colleagues like Atik Ahmed at Northwestern who specializes in that. And then they use those stem cells to study plasticity and how in particular, how these tumor cells adapt to chemotherapy. I mean, these cells are very, very sneaky, and they learn how to adapt even to really toxic therapies like temozolomide. And you can't improve the therapy until you learn how they adapt. Can you block that adaptation? Those are the sorts of things that Dr. Lothier is working on.
All right, we have a couple more minutes. So I think we can squeeze in one last question. So we're talking about patients donating tissue for research. Are patients and families ever provided updates on when that tissue is used or any research results that come out from their patient or their family member's tissue that was donated?
Not unless they specifically ask. They are allowed to ask, and they're also, by the way, if somebody just donates their tissue and then, a year, 2 years, 5 years later changes their mind and says, you know what, I don't want my tissue to be used for research anymore. All I have to do is call me up and say, look, whatever is still hasn't already been used, because if it's already been used, it's already been used. But if it hasn't yet been used, please destroy it. And we always do, although that's only happened, I think, like once in the last 7 years. As far as the postmortem series goes, that's a little different because now I've got more engagement with the next of kin and everything. And I always ask them, I said, look, the next time we're about to release another paper, do you want to know when that paper comes out? And, because of course, when we list our cohort, like, that one study I showed you, we had 33 cases and they were numbered 1 to 33. So they were completely anonymized. Do you want to know which case was your husband or wife or something? And sometimes they say yes, sometimes they say no, but a lot of them are extremely engaged in the process. I've had plenty of them at the microscope. We'll sit down together in my office and go over the slides. I mean, it's really quite an experience to be going over tissue slides of your lost spouse. But many of them do find a lot of healing in that, and they find a closure, and they want to know that their loved one's death had some meaning, and that it's being put to a good cause.
Thank you so much for sharing that. Unfortunately, that is all the time that we have for questions for this session. Thank you again for your time, and I'll go ahead and turn it back to Emily for the rest of the program.
Yes, thank you so much, Ambreen, for facilitating those questions. Great questions from the audience. And I just want to say thank you so much to Dr. Lothier and Dr. Herbinski for taking the time out to be with us today and sharing with us about laboratory and translational research. We will have a short 5-minute break right before our next session on development of new therapeutics. We'll see you back in a few minutes. Welcome back. I hope each of you took a brief moment for a break. I'm going to welcome back our chair, Dr. Vogelbaum, to introduce our next session and our next speakers.
Okay, thank you, Emily. Just want to make sure you're hearing me. Good. Okay. So
You sound great.
Super, thanks. The next session is entitled Development of New Therapeutics. And in this session, you'll learn about how a new therapeutic is created and initially tested. You'll also be able to explore the barriers that a new therapeutic must surpass before it can be used in a patient. Our two speakers are Dr. Nader Sinai and Dr. Amy Heimberger. Dr. Sinai is the J.N. Harburg Professor of Neurological Surgery at the Barrow Neurological Institute in Phoenix, Arizona. His specialty, neurosurgical practice, is devoted to patients with benign and malignant brain tumors, particularly those in high-risk areas. In 2018, he founded and now directs the Ivy Brain Tumor Center, a not-for-profit brain tumor drug development program. You may be interested to learn that one of Dr. Sinai's favorite activities is surfing, which is not an easy hobby to pursue in Phoenix. Dr. Amy Heimberger is the Jean Malnati Miller Professor of Neurological Surgery and Vice Chair for Research in the Department of Neurological Surgery at Northwestern University, and she is the Scientific Director of the Malnati Brain Tumor Institute at the Robert H. Lurie Comprehensive Cancer Center at Northwestern. Her research program focuses on immune therapeutic strategies for patients with central nervous system malignancies, and she studies tumor-mediated mechanisms of immune suppression. Prior to her arrival at Northwestern, Dr. Heimberger was at Duke and then MD Anderson, but her medical career started at Washington University in St. Louis, where she was a medical student when I was doing my neurosurgery residency there. She would often join our neurosurgery educational conferences, and I have to admit she was by far the brightest person in the room and still is to this day. Please welcome our speakers. Dr. Sinai will present first, followed by Dr. Heimberger.
All right. Well, thank you, Dr. Vogelbaum, for the introduction. It is true, it's difficult to surf in Phoenix, but Del Mar is only 5 hours away by car. What I will do is start off with a little bit of a discussion on the translational side of research, taking us from the bench, what we've heard about, into the bedside, and speak more specifically about the development of new therapeutics within that space. So no disclosures relevant to this talk. As Mike mentioned, all of our work is supported by nonprofit research dollars. As Dr. Horbinski mentioned, the technical capabilities of neurosurgeons like myself in the operating room has continued to expand over the years. And today, there's almost no region of the brain that can't be operated on, and no lesion in the brain that can't be removed, at least on the basis of what you see on a scan. So here you have a patient of mine, a friend of mine, who had a large glioma, which I don't need to train you to read scans, you can see on the left. And lesions like this can be so large that a single operation is not enough, and a combination of approaches are required to get to the back part of a tumor and then bring the patient back the next day to get to the front part of the tumor. And these operations can take many hours, as some of you are all too familiar with, but we can get patients through them. And for patients like this and others, the recovery can often go faster than expected. And the postoperative scans, which you see on the middle, look reassuring. But the problem, of course, is that there are little cells and things that you don't see on the scan, as Dr. Horbinski mentioned, that will then recur over time. And as that happens, the tumor reconstitutes, and increasingly, it becomes less and less of a surgical disease. So we say brain cancer, and gliomas in particular, are not a local disease, because what we see on the scan, even when removed, is not curative in most cases. Now, as many of you know, there are 4 sort of pillars to standard of care therapy today. The first is typically an operation done by someone like myself to remove everything that you can see on the MRI. And that's typically followed by a combination of radiation and temozolomide, which is the single drug that most patients get up front. And then for some patients, it'll then continue on with what are called tumor treating fields, which is an electromagnetic cap that some patients wear. The challenge, of course, is that we just have that single drug to rely upon. And in the last 40 years, there hasn't been much productivity in terms of new drugs approved for brain cancers like gliomas. We have a couple drugs that were developed in the 80s that are essentially derivatives of mustard gas. These are relatively toxic drugs that have some efficacy. A large period of time passed before we started to see new drugs FDA approved. One of them, most recently, bevacizumab, otherwise known as Avastin, doesn't have a survival benefit in gliomas, but can improve symptoms. And then even more recently, a targeted inhibitor, a drug called everolimus, was approved for a very, very rare subset of patients who have something called subependymal giant cell astrocytoma. These are typically children. And for these tumors, this drug is extremely effective, but it doesn't apply to the more common patients. So that leaves us with temozolomide, which today is still the single most effective medical therapy for gliomas. Now, why is it that we haven't had more success in developing new agents? Well, you've already heard some examples of how the biological challenges in this disease are particularly unique. This is a karyotype, which is a picture of all of the chromosomes in a normal cell in your body or mine. And this is a similar picture of all of the chromosomes in a glioblastoma cell. So you don't have to be a biologist to understand that this is an extremely convoluted disease. And this doesn't even touch upon what Dr. Lathia mentioned, which is the difficulty of getting new therapies into the brain. But beyond the science, what we know is that most drug development today happens by the biotech and pharmaceutical industries. And those industries are driven by market forces. And those market forces point them away from brain cancer. And in fact, it's fairly easy to predict where new drugs are going to be developed and approved by the FDA on the basis of the efforts from this kind of set of market forces. And so it's not a surprise that if you look at improvements in cancer survivability over the last 5 or 10 years, you see large market diseases like lung cancer and skin cancer having significant improvements, and brain cancer not so much. Now, this isn't to demonize at all the biotech or pharma industries. They're simply doing what makes sense for their business model, shareholders, investors. But it is to explain that there are differential levels of effort being allocated here. And at the national level, we've also seen a gradual sort of redistribution of federal support to support large-scale consortia efforts. 20, 30 years ago, we had multiple what are called consortia, which are networks of major brain tumor centers working together to develop new drugs, test new therapies. And over time, with the evolving landscape of federal funding, those consortia have gone from 3 to 2 to 1, and then most recently eliminated because of different priorities. Now, this is why organizations like the ABTA are so important today to fill in this gap left by some of the federal efforts. And alongside that, what often drives drugs to later stage clinical trials isn't always necessarily synced with the data. Because of the dearth of federal support and support in general in this field, many times efforts go where the money is. And so if there is funding to support a clinical trial, oftentimes that trial will happen with or without very solid data in support of going to that level. So this is part of the reality of today's drug development world. It's no different in other cancer types as well, but it is part of our challenge. And the final kind of component I would point to is that you're meeting today very, very determined individuals, and we all want to make a difference in this space. Sometimes, however, it's challenging when a program is working on an idea for many years or even more to pivot away and say, you know, this doesn't look like it's working. Let's go in a different direction. There are a lot of practical challenges there. This is part of a editorial by Lisa DeAngelis, who's a neuro-oncologist at Sloan Kettering, who points out some of the risks of just kind of pushing ahead at all costs. So you're going to hear today about different types of clinical trials, and I won't review the entire spectrum of them, but I'll focus a little bit on a certain subtype of clinical trials, which you could broadly consider as tissue-based trialing. So these are clinical trials where patients receive some sort of experimental therapy before some sort of operation. And then at the time of the operation, we try to ask questions about the patient's tumor to figure out if this experimental therapy is doing what we hope it is. So this is a little bit more specialized of a trial effort than, for example, a conventional phase 1 or 2 or 3 clinical trial where patients just get drug and we just follow them with imaging. One type is a phase 0 clinical trial. Another version of it is called the window of opportunity. And I don't think I'll get into the details of this for this audience, but there's even a hybrid version of these two called a PK/PD triggered trial. And so our program and several of the programs I think represented on this panel do focus on these types of tissue-based trials. And the general paradigm is something like this. As a patient, the patient has gone through standard of care therapy often and has a recurrence of their tumor. There's a genetic analysis performed of that tumor recurrence that matches them to some sort of experimental therapy. Maybe it's a drug, maybe it's a drug cocktail, maybe it's a drug device combination, but the patient gets exposed to that before some sort of planned operation. And at the time of that operation, we then test the patient's tumor and their other tissues and basically ask, what kind of biological effects are we seeing here? Is this thing working or not? And if we feel like there's some evidence that this experimental therapy is having its intended effects, then the patient goes on to get more of it. And if we feel like there's not, well, then we try not to waste the patient's time by going on what may be a futile therapy, and we try to get them onto a different clinical trial. So at our program, we call these trigger studies because of that kind of trigger of going on to therapy or not. Another point to remember is that in the conventional world of chemotherapies, the more chemotherapy you give someone, the more effect they will have. But today, most drugs that are developed are not chemotherapies. Chemotherapies are, in general terms, a toxin that basically poisons cells, and we hope that they poison tumor cells more than your normal cells. But today's drugs are what are called often targeted inhibitors. And those are targeting molecular mechanisms where sometimes more of the drug doesn't actually lead to more effect. So in many of the studies today, what we're not looking for is just how much drug can we have a patient take before they get too sick to tolerate it. We're looking for what is the optimal biological dose of the drug where we're seeing the maximal effects. And this is why for many newer clinical trials, it's important to look at the tissue of the patient's tumor after it's been exposed to the therapy to see where that maximum level is. So I'll give you an example of the journey through clinical trialing. A patient friend of mine, Patricia, who recently passed, and her family, she's a physician and was diagnosed with a glioblastoma. She went through standard of care therapy. She had a rare gene fusion that qualified her for a clinical trial in July of 2020. At the time, the tumor was on one side of her head towards the back. She went on this trial and had what looked like disease stability for several months, had a recurrence. And at the time of this recurrence, the tumor had changed itself, which is one of the challenges we face with this disease. The genetics that it had in July, it no longer had, and it had a new set of genetics in October. So that qualified her for a different clinical trial, which she went on, and then the tumor recurred on the other side. And here she went on an additional trial, this time targeting something that helps radiation effects. And then finally, she had a first in human clinical trial called sonodynamic therapy. So Patricia's journey began in October 2019. And for 9 months after standard of care, she had no recurrence. And then she went on a series of clinical trials, taking her from one portion of the year to the other and constantly shifting gears, trying to show this tumor different looks so that we could keep it at bay. And ultimately, she passed away just recently after almost 3 years of dealing with this. And I think that if you ask her and her family, they felt like these clinical trials gave them additional lifelines and windows of hope as they tried to move through from one experience to the other. Now, this is one thing that I would want to really emphasize to the audience, is that clinical trials are about creating this hope, and it is important not to lose them. This is the experience of
Nick
another
was,
patient
a natural born
and her parents
debater.
going through
I remember
this
when he was
kind of
about 2
experience.
one time, I thought, I'm losing an argument with a 2-year-old. This is not right.
Nick has always had a great sense of humor. He has enlivened the household with his exuberance.
Some things I'll love to be able to do in my free time is be able to get out into nature. whether that's hiking around the Phoenix Valley anywhere or spending time in Flagstaff or in the mountains in the state. I knew something was wrong physically because I'd been dealing with a lot of symptoms of nausea, migraines, and headaches. The headaches and migraines were 5 or 6 days a week. So this would be, I'd have a lot of problems dealing with lights during the middle of the daytime and just kind of would totally shut down and did not feel good overall. When I was diagnosed with a brain tumor, when my primary care physician said I had a brain tumor, I had kind of blanked at that moment, or those kind of, you don't know exactly what to think or how to process it.
So this is the tumor that Nick had, and I don't need to show you where it is or why this is bad, but he went on through treatment, and
You don't
his
look at a
parents will
healthy
tell you a little bit about that.
23-year-old and think cancer.
The idea of a brain tumor was nowhere on the radar. It was nothing in our family experience or our friends' experience, and so we were completely surprised and shocked by it when the diagnosis came down.
He had an aggressive operation over 2 stages, during which we removed all of the tumor, but we knew that there are microscopic cells to these tumors that we can't see. And so those were targeted with radiation in the hopes that they would respond and not allow the tumor to come back.
Nick was in the hands of as professional a group as was possible, and that the outcome was going to be as good as it could be, and everything was good for 3 years.
The day after I graduated from San Diego State University, I had met with my oncologist out there, and there had been a regrowth in my tumor.
We knew immediately that Nick was not going to have a lot of different types of options, certainly not options designed for this kind of tumor. But we had just opened one of our phase 0 clinical trials for this exact indication. The next step is to see whether the genetics of his tumor were a match for the genetics targeted by this drug. And so in Nick's case, he demonstrated all 3 biomarkers, and then we knew that he was eligible for this study.
Sounded like a really appealing approach at the time, or a very innovative approach to make sure that medicine is personalized, is working, and know that the medicine I'm taking is just for me.
So Nick went on to get this drug and continued on to the trial. He still today hasn't had a recurrence. There's a lot of individual results within our space, and you will always meet patients that have unusually good or bad responses to things. It's important to remember that individuality means that what happens with one person may not necessarily happen with another person, but this is part of our challenge, is sort of customizing our therapies. So in the last couple minutes, just to point out another type of approach that feeds into drug development and therapeutic development. We talked about these 4 modalities, and there are others in development. One is called sonodynamic therapy, which is a drug device combination that involves giving a patient an inert drug and then letting that drug get metabolized in the patient's brain tumor and being targeted with non-invasive, non-ablative ultrasound. And it's the convergence of this ultrasound with the drug that leads to the cytotoxic cell death effects on the tumor.
Phase 0 clinical trial. This is the first in human study. So sonodynamic therapy is a combination of a drug and a device. It's non-invasive, doesn't require an operation. A patient with a malignant brain tumor gets this drug the morning of the procedure, and then there is a frame placed on the patient's head. It's fixed to the patient's skull. It's done with local anesthetic, so it's painless. The patient doesn't feel any discomfort. And then following that, the patient goes into the MRI scanner and gets attached to the sonodynamic therapy device. Neuroradiologists, radiation oncologists, and neurosurgeons at the IV center work together to identify the target and then deploy the MR-guided ultrasound to actually target those areas with the focused ultrasound energy. The ultrasound itself is harmless, but it's the convergence of that energy with the drug byproduct that really leads to the cell kill that we'd like to see in the patient's tumor. So for us, this is exciting new territory that's been unexplored to date, and this is the first time for any cancer type where sonodynamic therapy has really been investigated in a patient. 4 days from now, the patient will go to the operating room for an operation that was already planned to remove this recurrent tumor. And when we remove the tumor, we'll be able to see exactly what effects we've had from this particular trial in this particular patient.
So as I mentioned, a critical component to a lot of drug development is figuring out what's happening in the patient's tumor. So here you have a patient that got a first-in-human therapy, have the tumor resected en bloc like you see there. And then we're able to look at the entirety of the tumor from one side to the other and basically go slice by slice and figure out what's happening in this tumor cell in response to the areas that we targeted and how does it compare to the areas that we didn't target. So I apologize for running a little bit over. I want to thank the ABTA as well as all of our folks at the Ivy Center and of course the audience for their time. And I'll turn it back to Amy and Mike.
Thank you so much, Dr. Sinai. That was really impactful. We're going to hand it over to you, Dr. Heimberger, and give you remote control.
Okay, so let me just go back. So what I do is a lot of translational research. And part about doing translational research is actually interfacing with companies in a variety of different ways to actually implement new therapeutics in patients. So this is the clinical translational pipeline. It starts out with preclinical R&D, and that typically means target discovery. And then you have to devise a drug. It could be an immuno-oncology drug. It can be a targeted therapeutic, some of which have been touched upon by Nader. In some instances, you need to work through whether there should be a companion biomarker, in other words, to identify the correct patient to go on that therapy. You also need to think through the priority of that particular drug. In other words, what indication you want to move it into. And then you have this whole continuum of going from phase 1 and window of opportunity and then to later stage clinical trials. Typically what has happened is that academic centers are the ones that do a lot of the target discovery and maybe some of the initial drug discovery. And then that typically gets handed off to pharmaceutical companies. But that traditional model, as I show you with the top arrow, has really sort of evolved. Go back, Emily. That has really sort of evolved now to a partnership model, to philanthropy, thank you, to government, philanthropy, academics, and industry. And in fact, there's an increasing movement of pharmaceutical companies to actually interface with profiling companies to do target discovery and then work on development of various agents. So every step along the way, you have to finance it. And every step along the way are critical, what's considered go and no-go decisions as you're developing a therapeutic. So criteria for CNS drug development. It's fairly easy to sort of go after targets in what we call liquid cancers, because a lot of those cancers have very homogeneous, in other words, all the cancer cells are very similar to one another in liquid cancers and some other types of cancers. So that when you apply a targeted therapeutic, most of the cancer cells die or are targeted. In the case of glioblastoma, one of the key confounders is that you have a cell and right next to another cell is not the same targets. And in fact, when you start looking at frequency of expression, many of the targets that are actually been discovered in glioblastoma, only a small subset of those cells actually express. And so this inevitably is a key attribute for why you see the inevitable recurrence of glioblastoma. When you're developing a CNS drug, you really want to look for a potential target where it's very commonly expressed and specifically on the cancer cells. There are some targets that could be with the associated stroma, like infiltrating immune cells or certain other cells, and there may be therapeutic benefit to those. But clearly you have to go after the cancer cell. So when pharmaceutical companies or those who do translational therapeutics are looking, at whether they want to do a go or no go with regards to target, they frequently want to see commonly expressed in most cells within that cancer. In many instances, our clinical trials are done at the time of recurrence. There's a group that obviously do newly diagnosed, but the vast majority are done when patients fail standard of care. So if you have retained expression of that target, that's obviously a benefit for a pharmaceutical company that's developing a therapeutic. Most folks want to understand how that particular target works. That informs combinatorial therapy, and it also informs potential biomarkers. If it's a mechanism of resistance, that also goes into the sort of impact at the time of recurrence. As far as the target, you want to make sure that the target has a relatively specific expression in the tumor versus on normal cells. Really desirable targets are exclusively expressed in the cancer. That would mean that there's a low chance of toxicity or minimal toxicity on normal cells as side effects of the drug. And then one of the key things that goes into competitive landscape and sort of understanding market share and whether you're actually going to have to do a biomarker, what's considered a companion biomarker, is the frequency of expression within an indication. And let me give you an example of this. You'll be like, oh, well, you already touched upon that, the first one with regards to frequency of expression in cancer. Within glioblastoma patients, you might have, let's say, 1% of those glioblastoma patients that, let's say, have a specific, let's say, NTRK fusion alteration. So if you enroll 100 of those patients on a targeted therapeutic and only one of those patients has the actual target, then that means that you have essentially 99 patients that have been exposed to the risk of that particular therapeutic and only one potential patient that benefited. And as such, because most of the time we generate data that shows median survival, in other words, the average response in a cohort of patients that are enrolled, then that would have been deemed as a non-successful clinical trial. So that is how come that you have these initiatives where you do molecular profiling or companion biomarker in the development of a translational therapeutic. Then you have to think about the actual compound properties. So the drug needs to actually hit target. That seems fairly obvious. You need to have that compound actually selected to the target. So a lot of the drugs we previously used are pretty dirty. In other words, they target many other sort of targeting pathways, or they target processes such as proliferation. And that's how come you have hair loss or diarrhea, et cetera. It's because it's targeting proliferating cells that are normal. We like to see anti-stem cell activity in some of these compounds. It would be desirable, but it's not an absolute. And additionally, you really want their drug to really hit that target, like cancer, at a much, much higher ratio than normal cells. So if it's a fairly short or fairly small little interval of how toxic the drug is to the cancer versus to the normal cell, then you're going to experience a lot of toxicity when you actually give that drug to the patient. With PK, that means that when you actually have a drug that hits the target in vitro, you want to be able to achieve that concentration when you actually administer it, either IV, orally, or however the route of administration has been set for that drug. And in our particular case, what's unique to us is the fact that we do have issues with regards to CNS penetration and delivery. And so now there's a great deal of interest in a variety of delivery strategies, everything from blood-brain barrier opening strategies to ultrasound strategies to some dynamic therapy, et cetera. And then it's how rigorous these actually demonstrate a therapeutic effect in a variety of preclinical models. And so usually the more preclinical models you can demonstrate therapeutic effect, the more confident we kind of are with regards to go decisions in moving it into clinical trial. So what I want to do is I want to give you one example of walking through this whole continuum. And up in the upper right-hand corner, you're going to see where it goes along that pipeline. I'm going to give you the starting year that those investigations actually were triggered, the mechanisms of how something like this was funded, and then I'm going to give you the sort of running tab for how much this actually costs if you're going to do it de novo. So what I'm going to show you is a drug we developed for STAT3, the signal transducer and activator of transcription 3. This is a key driver of cancer. It is common in many different types of cancers, and this is part of the reason why it actually got go development is because it's not exclusive to glioblastoma. It can be induced by a variety of factors. It can essentially regulate tumor proliferation, survival, migration, and invasion, and it drives angiogenesis. It's a negative prognosticator for survival. It's commonly expressed in high-grade gliomas. It mediates what's called malignant transformation that was modeled in gem models of mice, and it maintains cancer stem cells. And you can see that this target was actually, we started describing this back in 1994, and essentially a variety of investigators, R01s, which usually is about $250K per year, essentially paid for a lot of sort of this basic biological understanding. It also is a key regulator of tumor-mediated immune suppression. So when STAT3 becomes activated in immune cells when they infiltrate into the tumor microenvironment, it essentially turns off their ability to kill cancer cells. And so essentially it turns off antigen-presenting cells, it induces immunosuppressive macrophages, it turns on immunosuppressive Tregs, and it triggers the transcriptional regulation of a whole slew of immunosuppressive cytokines. So again, the way this was studied was through NIH and R01 mechanisms. So one of the drugs out of 300 that were initially devised to go after this target was the drug WP1066. So this is a small molecule inhibitor. 300 different drugs had to be created and optimized to ultimately pick this one that was moved forward. And this was actually driven through a variety of institutional funds at MD Anderson, as well as philanthropy. And this component was about $900K, so almost $1 million to come up with an initial lead test article. So this drug is a derivative of cathaic acid. It's found in beehives, so it's a natural drug, but then it was modified through sequential cycles. It has a high degree of blood-brain barrier penetration, but this actually became a challenge for the formulation. It markedly inhibits the translocation of STAT3 into the nucleus, and it markedly disrupts stem cells. Pilot toxicity data, again, this was now funded under a SPORE mechanism, and you can sort of see the timeline here, 2008, showed that it was well tolerated in a variety of animal models. And then additionally, it showed a marked increase in a wide variety of mouse models, preclinical mouse models. So all of this is published where it showed in vivo activity against gliomas, AML, melanoma, squamous cell, renal cell, non-small cell, gastric, and breast. And again, we're still at the level of sort of NIH R01 funding here, where all of this data was generated. It can reverse T cell dysfunction from glioblastoma patients. Again, this is again in the preclinical realm where we're working through biomarkers, and some of these actually were incorporated in the clinical trial. Here's some additional biomarker work where it blocks immunosuppressive immune cells, just sort of showing you some data that was published. And then this is the scorecard for this particular drug. So one of the things in our particular program we think about is while we're developing a variety of different types of agents, everything from CARs to immune modulatory aptamers to BiTEs, et cetera, is we actually rank our program based on that criterion that I just showed you as a way to prioritize what we really want to move along the continuum. And that's because that continuum is extremely laborious and it's extremely expensive. So here's what it takes to work on a formulation. So in order for us to now move this forward into patients, we had to actually devise a formulation that would be suitable for clinical trial administration. And now this comes into the range of philanthropy and mechanisms called the small business. Essentially, these are grants that are given to new codes through the NIH. And this required about a price tag of about $500K, where we combine the funding to work on what's called the good manufacturing practices, as well as the regulatory package for the formulation. So this adds up just to get this sort of formulated into clinical trials. And the way we did this was with that drug that's highly lipophilic, we coated it with methylcellulose so that essentially it had a more steady release into dogs. And this is the formulation that you actually see in the human subjects. So then at some point, you have to figure out how you're going to pay for the clinical trials. So you have to either be really, really savvy with VC, or you need to do some kind of construct of a patent or a licensing deal. And so essentially there's some key elements here. You have to be very careful with what you disclose in the public domain. So you have to have the patent filed before you actually start publishing this. So sometimes there's delays in publications. The licensing deal is really where you sort of generate revenue from your intellectual property. It's not the patent in of itself. You usually need to clarify what your role as a PI is going to be moving forward, because a lot of institutions have conflict of interest with regards to a PI that's holding substantial amounts of funding from a licensing deal. And additionally, you need to understand that the further along the pipeline that you develop something, a lot of times that's the bigger the deals that you see in various news releases. So for example, you may hear of sort of strategic alliances where somebody makes, $15, $20 million. Usually those have already gone into early stage clinical trials for that investigator. So this drug has to be clinically ready. So you have to have an intellectual property patent filed. You usually have to have a licensee granted, or you need to be very savvy in the VC. Still needs to be on patent usually to get VC involved. You have to have an IND. You have to have the commitment of whoever you license the drug to. And if you don't have frequent expression, you need to be thinking about that companion biomarker, and you need a clinical trial ready to go. So right here, this shows you what an IND looks like. This is how much goes into the actual documentation for developing one of these. And so what's in this packet? Stability studies, chemistry, manufacturing, and control, drug characteristics, good manufacturing practice. In other words, you have to have all your certificates of analysis and your release criteria. You have to have all your toxicity and your toxicokinetic analysis. Usually that involves at least 2 species, usually rodent and usually canine. Preclinical studies, you need to have a dose finding analysis. Usually the FDA, although completely acknowledge what Nader has already indicated, the FDA almost always requires you to do some kind of maximum tolerated dose finding in the preclinical realm. You need mutagenic assays. In other words, you don't want to trigger additional cancer in patients by giving an agent. If you're going to give it to potentially pregnant ladies, you need to also do teratogenic assays. And then you need that clinical protocol. And so this is not something I can do in my lab or Nader can do in his lab. Many of these components have to be done in accredited laboratories that the FDA recognizes. So you usually need to come up with either some kind of philanthropy, VC, pharma, some kind of funding, and a first end man. The cost for something like that is about $3 to $5 million. Now you can do a cheaper biological IND, but this is usually the going cost for a first in man. In other words, a drug that's never before been put in a human subject. So this was that drug I was showing you. So the phase 1 clinical trial costs, this is a really, really cheap, no frills kind of clinical trial. Usually 1 to 2 million is the cost to actually up and implement. We did 2 clinical trials, one at MD Anderson. We did the other one that's still active and ongoing at Emory and the pediatric patients. So we enrolled both adult and pediatrics. We actually did an accelerated titration design. We did a number of biomarkers, and the primary objective was to determine the maximum tolerated dose, safety, and tolerability of this drug. And these are the protocols numbers for the government clinical trials registration. So we reported out the phase 1 clinical trial. We had 5 dose levels. We went from 1 milligram per kilogram up to 8 milligram per kilogram. The methylcellulose coating of the drug tastes astringent. It's like taking aspirin by mouth. And so patients really dislike that. So really we got to a feasible dose of 8 milligram per kilogram. The pediatric patients, we are moving on to 16 milligram per kilogram and seeing if we can push a little bit further up. As far as the results, we saw marked inhibition of phospho-STAT3 in the peripheral blood. So this is flow data in the blue curve. That's before the patient started dosing with the drug. And then the green curve, you can see the inhibition of the phospho-STAT3 in the peripheral blood. And so remember, this has immuno-oncology properties in addition to direct cancer activity. So this was the progression-free survival and the overall survival. This is a small cohort of 8 subjects. We don't see a signal as a monotherapy as would be anticipated with most drugs. As a benchmark, there was 37 patients enrolled on the temozolomide phase 1 clinical trial, and there was only 1 objective response reported out in that clinical trial. Toby at Emory has 2 patients with radiographic responses. Both of those patients are with DIPG. Sorry about the little glitch there on the PowerPoint slide. And this is a patient that was wheelchair bound. You can see the large T2 flare at midline on this particular patient. The patient became ambulatory and then had a response for 5 months. And he has a second one as well, in which is shown also a clinical response and radiographic response to this drug. And so he has 8 enrolled patients so far, and he's at 8 and getting ready to move to 16. So one of the things is, how are you going to combine this with standard of care? And so we did some preclinical work where we combined it with radiation, because that's typically what happens after a patient gets a resection. We, in the preclinical mice, dosed this at 60 milligram per kilogram. That converts to 5 milligram per kilogram in human subjects. It's something called allometric scaling. And so that means that with that previous phase 1 clinical trial that I already showed you, that means we've already exceeded the dose that we're using in the mice. We're now back to sort of looking at priority and indication here. Again, we're back down to NIH and R01 funding at about $250K per year. What we found was when we dosed those animals, that we did a therapeutic window of opportunity analysis in the mice, we saw that this combination of a radiation as well as the drug, the STAT3 inhibitor, induced very marked dendritic cell and T cell interactions in the tumor microenvironment. And those don't normally happen in GBM patients normally. So this was a very nice sort of immunological effect with the combination. And as you saw, there was an increase in survival in those mice. So now this is what the phase 2 clinical trial looks like. So we have patients that will have the standard of care surgical resection. Those patients will be MGMT unmethylated, so they do not necessarily need to be treated with temozolomide. So they'll receive the combination of radiation and the STAT3 inhibitor. And then for those patients who did not get a gross total resection, they will also be treated with the radiation and the WP1066. And if they still would benefit from a clinical debulking, then we'll offer surgery so that we can analyze in that tumor microenvironment whether this combination induced those dendritic cell T cell interactions. And this is our go, no go for later stage clinical trials. So this includes, it's a phase 2, but it also contains a window of opportunity component, where we'll also be evaluating the concentrations of the drug in both the contrast enhancing and non-contrast enhancing component. So just to give you a little bit of an idea here, for each site, usually it's about a minimum of $1 million per site to get them up and going as far as the cost. And that includes things like nurses to monitor the clinical trials, the infrastructure, the submission to the IRB, et cetera. So really, the ABTA, National Brain Tumor Association, many of the philanthropic organizations have been absolutely pivotal in powering these clinical trials and being a huge advocate. So we thank our patients also for participating and being willing to, sort of participate in these kinds of studies. So I thank you. And I think that's it for me.
Well, thanks so much, guys. Ambrina, are you going to lead the Q&A now?
Yes, thank you so much for that great presentation. As a reminder, if you have questions that you'd like to submit, please type in submit it using the Q&A button on the bottom of your screen, and we'll answer questions as time allows. All right, so one of the first questions we have is, Dr. Lathia, you had mentioned if it's determined that there's no intended effect or benefit while a patient is on a clinical trial, then they will be transferred onto a different treatment or a different trial that will benefit them. How long does that time frame usually look like? How long do you wait to determine or make that determination that there is no intended effect or benefit?
I'm not sure who you were directing that at. You said Dr. Lafia, but we have Dr.
I'm sorry, Dr. Sinai. I'm so sorry. It's been a morning.
Not a problem.
Good catch.
Yeah, Ambreen, thank you for the question. Yeah, so I think what you're referring to are clinical trials where patients get exposed to a therapy, they go to surgery, and then we wait and see what the consequences of that therapy are before we continue them on therapy. So in our system, that takes around 7 to 10 days. As Dr. Heimberger mentioned, a lot of these trials have to be done in certain sort of federally validated laboratories. And we have a name for this called CLIA CAP certification. So in trials like that, you have to have a CLIA CAP certified laboratory that can do this work quickly in a rigorous way where you're making a clinical decision on the basis of the results. So these trials typically will require about a week for that to happen. And during that week, as many patients know, you're still recovering from surgery, you're hopefully out of the hospital, getting over things. And then shortly after the stitches or staples come out, that's when we have an answer with regard to whether or not it makes sense to continue on the therapy.
Thank you so much for sharing that. And Dr. Heimberger, you shared a timeline of how clinical trials can progress. Can you share or maybe give our audience some idea of how long it can take an investigational therapy to go from the lab to the bench side? I know that's a loaded question.
As I showed, some of this stuff, the target elucidation started almost 20 years ago for this particular example I'm showing you. If you've got big pharma, let's say you got BMS, Merck, somebody like that, they've got a lot of, funds and resources where they can drive this really very quickly, especially if it's a high prioritized program in their pipeline. And so they can expedite things. You think about the timeline of, I showed you a couple times NIH grant funding. Well, it takes about a year, even if you get a great score on that, to put that grant in and get the money actually through the door. So I think one of the things we hear all the time is, and certainly I am completely empathetic with patients with, what's taking so long. That was a key point of what I was trying to illustrate was you've got to get all of these things organized and you have to figure out a way to MacGyver the funds together to drive it. Because unless it's a key initiative with pharma, and they're a big pharma, a lot of the drugs we get are not necessarily from big pharma, they're from smaller companies. And so they're really cash poor and they can't really drive a lot of this stuff. And so they're sort of scrambling and trying to figure out the cheapest way they can actually do this because again, as Nader, I love that slide Nader showed, by the way, of, sort of where does the money go, and it goes to the big three, prostate cancer, lung cancer, and it goes to breast cancer because that's the biggest market share and patient population where, they're going to recoup, their investment. So I would say, really impressive from like target all the way to a drug. I would say, wow, maybe 7 years would be really, really fast. Because again, it's just the regulatory components in of themselves can be highly rate limiting.
If I can add to that, I think that, one of the challenges we've had in our field is that we get a lot of hand-me-down drugs for the reasons that both Dr. Heimberger and Dr. Sinai has outlined. That's where the initial investments go. And then when they're approved in another cancer, then we get to try them.
Yeah.
And many of our clinical trials have been exactly that. We just go ahead and try them. Part of what, Dr. Sinai has been doing is to actually step back and look more carefully. Do these even have a chance? Because many of them don't get into the brain, and that's why that work is so critical. And the fact, as he talked about, the funding that he has in his center really helps to accelerate that process. Because Dr. Heimberger talked about getting NIH grants, and it takes a year to get the money. That's if you have the grant awarded. And
yes, that's true.
for too long, our peer reviewers didn't understand that the idea that the drug has to get in the brain is a problem. And so, Dr. Sinai has been able to accelerate that. What Dr. Heimberger is talking about is starting de novo with a brain tumor specific target, one that drug companies are not going to be jumping on because that's not where they're investing things. And that is a very long process because we have to rely on so many different funding sources to be able to see it through. It's a remarkable achievement.
Thank you so much, Dr. Vogelbaum, for adding to that. Dr. Sinai, we have another question for you. You had shared the story of Patricia, and you mentioned that her tumor had changed. There were some differences in the genetic makeup. How often does that happen? Is it common for a tumor to change its molecular profile?
Yeah, I think we basically assume that happens all the time. This is one of the challenges of this cancer type, is that for other cancers, largely those cancers remain the same. One cancer may evade a therapy with a minor change to itself and then get treated with a second line therapy. So for example, things like melanoma, breast cancer, there are first line, second line, third line therapies that have all been proven effective. So you ratchet up the pressure on the tumor because it's not going to change all that much. But with gliomas, they're much more dynamic. And the more we look, the more we see these changes happening at every level. So this is why it's very important for patients to receive multiple different types of treatments over the course of their therapy to constantly put different types of pressures on the tumor and hopefully keep it contained as it tries to adapt.
And a follow-up question to that, how often is molecular profiling redone? Is it at the time of recurrence, or is there a set amount of time that you would look at it again?
Well, there are no hard and fast rules on molecular profiling, but I would say for most major brain tumor centers like ours and Northwestern and everyone else represented at this program, I would imagine it's done every time. Basically, whenever there's tissue re-resected, what that means is that the tumor has probably recurred. And so you have to reassess everything at that time, including the genetics.
Thank you for providing that clarification. Another question for you, Dr. Sinai. You talked about sonodynamic therapy. What tumor types are good candidates for this type of therapy?
Yeah, I highlighted sonodynamic therapy just because it's what's called a first-in-human therapy, and there are many different first-in-human therapies in development for gliomas. But like all first-in-human therapies, there's much more that we don't understand than do. So I don't think that we could say right now whether it has applicability to any type of tumor or all of them. We're trying to figure this out. And this is one of the frustrating aspects of therapeutic development in our space, is as much as we want to go very quickly, there are still fundamental questions. So with sonodynamic therapy, I mentioned that you're giving ultrasound energy, but how much energy do you give? For how long do you give it? Where do you target it? How many times can you do it? Those are all questions that we still haven't figured out. And so without that information, it's hard to know how you apply it.
All right, another question we have here, both you, Dr. Sinai, and Dr. Heimberger, you touched on toxicity. Can either of you maybe jump in and explain what you mean by toxicity, what that threshold is, and if it differs from treatment to treatment?
Do you want to take that, Amy?
Sure. So the toxicity profile is going to be dependent on the drug or the therapeutic agent. Toxicity is any undesired effect. Most of the time for patients, undesirable effects for most sort of anti-proliferative cytotoxic agents are things like hair loss, vomiting, diarrhea, those kinds of issues, bone marrow suppression. Those are all sort of, side effects where it can limit how much of the drug that you can give. It depends a lot on the drug and the route of administration. So let's say, I'll give you some other examples. So let's say you're going after EGFR, epidermal derived growth factor. You can have problems when you target that skin toxicity if you were to administer it systemically, because your therapeutic could also target the skin. You can also have ophthalmological toxicities as well. Some of this can be overcome by local administration by neurosurgeons such as Dr. Vogelbaum. The issue comes in how many times can you actually administer it directly into the brain to bypass some of the systemic organs. What's the best way of doing that? We've previously done things like convection enhanced delivery, but there's limitations to the distribution, backflow of the catheters, et cetera. So toxicity just means that you have an unattended side effect of the drug that is undesirable for the patient. And it could be manifested in a wide variety of different ways. Nader, did you want to add on to that?
No, I totally agree.
All right.
Bree, can I ask a question that came up in the Q&A? Because
Yes.
I think it's very relevant here, and that is, you two have both talked about very, very early stage specialized clinical trials that really hold a lot of promise. And one of the questions is that patients don't always hear about these from their local treating physicians. How do they learn about this? How do they get to be part of these clinical trials?
Yeah, go ahead, Amy.
So one fairly easy resource that any patient can sort of access is there's actually a clinicaltrial.gov. In order for any of us to run any clinical trial, it has to be registered. And so there's a little search page. If you just put www.clinicaltrials.gov, you can type in glioblastoma. Now that's going to give you a huge list of potential agents. And so some of them are terminated, which means they're no longer active. Some of them haven't started yet. It'll say not yet recruiting, but then you'll see a whole list of active clinical trials. That'll give you some general idea of sort of how many are ongoing. To sort of wade through that, one of the things I typically would recommend is maybe even just a telemedicine call with a major center that really has sort of an understanding of the entire sort of, let's say, national or worldwide portfolio of sort of what's going on. Some of us know that there are signals in some of the clinical trials and may have preferences. Maybe there's very good preclinical data that we feel strongly have, a high potential. And so a lot of times I get calls from folks that say, what clinical trials are you thinking, would you, for this particular situation, would you think mom or dad should go on? And, sometimes I'm happy to sort of say, these are sort of the things that, I think right now that have some either preclinical compelling data or maybe some early phase clinical trial data. But I would say talk to somebody who's actually been around the block a little bit. Mike's been around the block, Nader's been around the block. We sort of know the various clinical trials that are happening, worldwide and nationally. And so a lot of times those folks really have a good sense of sort of is well-based in science. Nader or Mike, did you want to add on to that?
No, I think in general, you guys, everyone in the audience should try to go along two routes. One is you doing your own diligence. So going to clinicaltrials.gov is one way to do that. So you inform yourselves. And the other is to use the specialists around you. And so getting second or third opinions is never wrong. All of you probably have a treatment team that constitutes at least a neurosurgeon and a neuro-oncologist, and maybe also a radiation oncologist. And everybody wants the same thing. We all want for patients to do better. So it's never wrong to approach someone else for a second, third opinion and just get a different perspective.
Thank you so much for covering that very important question. It's one that we often get on the care line as well. And I encourage anyone who's looking for more information on clinical trials to please reach out to the ABTA care line. And I'll put the phone number in the chat as well as our email. But unfortunately, that is all the time that we have for questions for this session. Thank you all so much for your input, and I'll go ahead and turn it back to Emily.
Thank you.
Yes, thank you so much, Dr. Sinai and Dr. Heimberger, for that wonderful session and for sharing with us how a new therapeutic is created and initially tested before used in a patient. All right, everybody. So for those of you who do need to take a break, this is now your opportunity to do so, to stand up, stretch your legs, use the restroom, or grab a snack, a beverage at this time. We invite the rest of you to stay on to hear from our sponsors who will share about what resources they offer to patients. We will resume the program in about 15 minutes for our next session on clinical trials, partnering with you to advance research. Thank you so much. See you soon.
Novocure has been a part of the oncology community we've served since the inception of the organization. And over time, the environment has changed, but our commitment to our patient advocacy groups that we work with has not. It has actually grown.
What really fuels us, or what gives us that motivation to go that extra mile, is reflecting on patients that we've heard from, patients that we could be impacting. It's really our source of energy and drive to help move the organization and our therapy forward.
We want to be able to help and support as much as we can. And part of that is you build up trust. So we always want to live up to the trust that has been placed in us.
In the first years of our marriage, 30 years ago, we would say, we're gonna be the cutest old people you've ever seen. And now we have to kind of rethink that. I think we're very cute now.
That story makes it real for us. It ties it to a life that we're impacting. It ties it to a caregiver who is taking care of somebody that they love.
It takes work to be present and appreciate what you've got. Since the diagnosis, I work at that much more. And I think that's a really powerful thing.
We actually get the privilege to meet many patients, and we never take that lightly. So we're always doing our best to constantly find out what their needs are, how we might support them better, how we can work with our advocacy partners to help the needs that they're trying to address in the community.
It's not just all about the person receiving the caregiving. It's also about them, the caregivers.
It's nice to have somebody with you that can hold your hand and just be there for a shoulder to lean on. I'm just here because she's the one that I love, and it's just what you do. There's no caregiving. That's a job, and this isn't a job.
I think it's important not to forget who you are, you yourself as a person.
I tell him every day I appreciate him, and I don't want to take him for granted.
I can't remember her not being in my life, and I cannot envision her not being in my life. And she will always be in my life one way or the other. I would encourage other people not to give up, to fight.
For me, I've decided that to define a day as a good day, have I been useful? Have I been creative? Have I learned something? If I can say yes to just one of those questions, then it's been a good day. And then I can have that attitude for the people I love.
We live in a very rural area, and you can almost count on one hand or maybe two how many radiation therapy centers there are. There are not very many. So I think for us it's been really helpful being in a rural environment that we don't have to worry so much about when or where these people are going to go for their radiation.
This gives them an opportunity to get their radiation kind of in a one-stop kind of fashion versus multiple appointments and trips to Greenville.
One of the things I was really excited about initially with GammaTile, because it's brachytherapy, it's actual radioactive sources delivered to the surgical cavity, it's inside out.
GammaTile is different from traditional radiation because you don't have to come in day after day for repeated treatments. It's also delivered directly at the time of surgery.
In the patients who have GammaTile, the surgery process is not really much different. You put the tiles in, the radiation starts immediately, and they don't come back and forth like we're used to for their conventional radiation. And many of them had 30, 35, 40 treatments. They don't have to do that.
This is something that I can hold in my hand and say, this is how it's delivered. You know, the radiation beams are inside the tile and they're physically implanted at the time of surgery.
Having an actual radioactive source, the dose fall off is so fast from that surgical cavity that large parts of the brain that otherwise would get radiation from external beam techniques get no radiation at all.
It's a great option for those patients who, you know, have already had radiation and failed it. It gives them an extra option.
Welcome back. I hope each of you were able to take advantage of this short break. I'm going to hand it over to our chair, Dr. Vogelbaum, to introduce our next session and speakers.
Thank you, Emily. So our next session is entitled Clinical Trials, Partnering with You to Advance Research. This session will provide an overview of what constitutes a clinical trial, how they're developed and run, the distinction between therapeutic and non-therapeutic trials, and why clinical trials can be seen as an essential part of care. However, we're going to begin first with our patient voice. I think this is a special part of the program. You've heard a lot from doctors and scientists, but it's not that often that you get to hear from someone who's been on the journey that many of you are on. Joining me today is Larry Thomason, who was diagnosed with melanoma in 2014 that metastasized to his brain in 2016. For those who are not aware, prior to the mid-2010s, the development of melanoma brain metastasis was an almost universally fatal turn in the disease. But then effective targeted therapies that actually reach the tumor tissue in the brain and subsequently immunotherapies have changed the course of this disease. And these therapies were first evaluated in clinical trials. We welcome you today, Larry, and thank you for your willingness to share your experience with the clinical trial that saved your life. Welcome, Larry.
Yeah, good afternoon. Can you hear me?
So, yes, we can hear you just fine. So perhaps you can talk to us about that point at which you developed brain metastasis and what were the treatment options that were offered to you and how did you decide to pursue a clinical trial?
Yes, we went through, after the original surgery for melanoma, we went through two series of scans and they were okay. And then the third one, we had a huge surprise and that we had, I had so many small melanoma in the brain they couldn't count or wouldn't count. And I had 5 in the frontal lobes, 3 which were already severely hemorrhaging. So we were directed, obviously, I'll just skip forward all the way to your colleague, Dr. Forsythe there at Moffitt. And he covered for us the clinical trial coming that was in progress, Checkmate 204. And that was after we had also met with Dr. Erlew, who had indicated there were several standards of treatment, but due to the extent and the magnitude of where I was in the process with the really severe stage 4, none of them were viable. So at that point, when we met with Dr. Forsythe, he covered the Checkmate 204, of which we were very fortunate. He was second chair for the entire Checkmate 204 program. And I know you're very familiar with Dr. Forsythe. And with that, we had a clinical trial coordinator, Anise Solis, was there. And she had a 48-page document that she covered the key points of. And of course, for us, there was really no decision. This was binary. There was no, this was a life preserver for us. So the only decision we had to make was, could Dr. Forsythe, would we qualify for the trial? So that's what the way we entered into the trial.
So that must have been a very scary time for you. How did you deal with your fears? What resources were available to help you with that?
Well, I think, just to go back a bit, I was totally asymptomatic, stayed asymptomatic. So from the fear standpoint, the only fear we had was not being approved. I wasn't, it didn't even enter our consciousness to be afraid of the trial, because it was the life preserver. So from there, we studied very carefully. That was on March 1st, and we only had a few days to go home, and we had to go through, obviously, all of the, make sure that all of our affairs were in order because of the seriousness of the situation. And then we came back on a Monday, which would have been like March 7th, I guess, 6th or 7th. And we spent 2 hours with the trial coordinator who went through the entire document. It wasn't a legalese document. It was a well-written, understandable document, covered a whole lot of ground, including the possible adverse events. And it was so specific was that if we had an adverse event, we knew that there was a category of 8 or 10. It was also specified in that document exactly what the follow-up treatment would be to address the adverse event. So we got that out of the way. We actually started the trial on March 10th then. And that was, you know, as I think back, in some ways we were very fortunate. In many ways we were very fortunate. But we didn't really do a lot of consternation, frankly, because our situation was so unique. We didn't have an alternative other than the trial, and that had been made very clear with Dr. Erlew, who said, if you do nothing, you've got 6 months, and most of that in hospices. So we were dead set and eager to get into the trial and get the trial started.
So you must have hospitals that are closer to home than Moffitt. What you know, it's not just a place. It's not just Moffitt itself. It'd be like any major tertiary care center. What made you leave the hospital near home and go to a specialized center? Yeah, well, back up when I had the first surgery, when it came back, a phase 3B melanoma. We're in Destin, Florida, 6 hours away. My dermatologist had connections with Moffitt, and by the time when he told me the results of the biopsy, he already had me an appointment at Moffitt to go and get the surgery. I ended up with 2 surgeries. So all of the information, everything was there. And the way he put it to me, if this was me or my family, there's only one place I would go for melanoma, and that's Moffitt. And that was the impetus of how we got introduced to it and gave us the confidence that that's the only place we wanted to be. And I suspect he was thinking, you know, Moffitt, there are other major medical centers because we have the clinical trials. I mean, that's really the difference between, you know, what someone can do in their local community versus, you know, one of the major centers you've heard about today. Yeah, well, we, you know, we had some time between the first meeting with Dr. Erlew and Dr. Forsythe. We went through and on the internet and looked at various facilities and centers, and of course there were 5 or 6 that were obviously good, but Moffitt was the closest. Moffitt was highly rated, maybe top rated, and that was, again, we had no reason to go somewhere we weren't familiar with. So now sometimes patients are very nervous about going on a clinical trial. It seems like an experiment to them. Can you speak to that fear? From the standpoint, you know, my background was in mathematics and statistics. And again, from March 1st to March 10th, I was on the internet. I'm not sure I slept very much, looking at various trials. I read up on every checkmate trial that had been completed or in progress. So from the standpoint of looking at data, there was a lot of data available. And we tried to absorb as much of that data as we could. And that with the 48-page document we were looking at, we had a lot of confidence. And another point of confidence, when we were first introduced to Dr. Forsythe, when we were leaving, I said, Doc,
what's my probabilities here? He said, well, Larry, I've got good news and bad news. He says, the bad news is you have the most aggressive form of melanoma we think we know of. He says, the good news for you is in the phase 1 trial, it does appear the more aggressive the form of melanoma, the more easily it's unmasked by the Yervoy and Opdivo immunotherapy. So he said, that's a big plus for you. So again, that was we felt like we had grabbed the life preserver at that point. So we went into it optimistic. And, the other thing that's very unusual, for all of the problems I had, I never had a headache. I never felt sick. I was walking 4 and 8 miles a day. I was playing golf when I got the diagnosis. I was doing the same thing after I got the diagnosis. We had a 10-day period there, obviously was a lot of things going, but our lives, we're so fortunate. Our lives, the way it all fit together, almost didn't change other than the mental, there was mental agony, of course, but we were from day one, we had optimism. And that's the way we proceeded forward.
Well, thank you so much, Larry. Really appreciate you sharing your story and your journey. And you're going to be joining us again later on our roundtable when we'll be talking about some of the challenges to doing clinical trials. So thank you. And I'm going to introduce our next panelists.
Okay, thank you.
So now I'm going to welcome our next speakers, Dr. Yoshi Umemura and Dr. Suzanne Chang. Dr. Umemura is an assistant professor of neurology and co-director of the University of Michigan Medicine Multidisciplinary Brain Tumor Clinic, as well as director of Neuro-Oncology Division and Clinic at the University of Michigan. Her career focus is in developing therapeutic clinical trials through a collaboration with translational and basic scientists, like the ones you've heard from, and across other clinical subspecialties to facilitate bench to bedside studies effectively to bring novel therapeutics to patients with brain tumors. However, few in our field are aware that Dr. Umemura spent 6 months after college studying contemporary and modern dance at the École Nationale de Musique et de Danse in Paris, France. And for those who couldn't understand my butchered French, that is the National School for Music and Dance. Dr. Suzanne Chang is a neuro-oncologist at UCSF whose research interests in clinical trial design and conduct, as well as imaging biomarkers. She's the medical director of the supportive care programs to enhance quality of life for patients and caregivers. I've known Dr. Chang since the beginning of my career. I actually have a picture from 25 years ago from a symposium that she attended when I was at the Cleveland Clinic. She's been an inspiration to me, but also to most everyone in our field, as she has been a leader in almost every aspect of what we do in terms of patient care, scientific and clinical research, and also the development of supportive care networks for patients and families. Dr. Chang is also quite humble, so if I say more, I'll just embarrass her, so I'll leave it at that. Dr. Umemura will present first, followed by Dr. Chang. Yoshi?
Thank you so much for the personalized introduction. And thank you so much for the invitation to present amongst exciting speakers today. So today I will be focusing on, I'll be focusing on kind of basics of what clinical trials are, what they are, and why do they even matter. And I reckon, I heard that one of my patients' family may be joining today, and some of my patients may have heard some of the talks, the things I talk about today in clinic. And I really want to give you an overview of basics of therapeutic clinical trials and why they matter to you. So the things I'll be covering would be how are these clinical trials conducted? Again, why do they matter? And the different types of therapeutic clinical trials. So going back to the basics, you've already heard a lot about clinical trials today or clinical studies, but what clinical trials are? They are essentially research study, medical studies to discover ways to how to find out what kind of brain tumors you have, how to treat them, when you're being treated, what kind of supportive therapies are helpful. Are there ways to improve diagnosis? Are there ways to improve prevention of disease? And these are all different kinds of trials that you can have. And essentially specific to brain tumors, oftentimes our clinical trials, the ones that we focus on, tend to be determining if a specific treatment is, if they are safe, if they are feasible to be done, if they are effective, or if they're even better than things that we have right now. And sometimes there are treatments. Dr. Vogelbaum mentioned hand-me-down medication earlier in the Q&A. Sometimes we have medications that may be working for other cancer, and are they going to work for brain tumors? Are there any medications that are used for other diseases with different mechanism of action? Are they beneficial for brain tumors? And we have a clinical trial like that in University of Michigan. And there are clinical trials. Some of you may be part of national trials comparing one treatment to the other if, when there is no consensus that one is better than the other, looking into one if one is superior or the same. And those are all different kinds of clinical trials that can be done, and they're designed differently. Clinical trials, when they are done, they're not done at someone's whim. They are reviewed carefully, not just by FDA, but also by institutional review board that reviews before the trial study is even conducted, not just for clinical trials, but all research, human research considerations about ethics, safety, feasibility, and if there is fund to adequately cover the study, because what you don't want to do is just start a study that cannot complete. Ongoing basis, IRB will also be reviewing the integrity of the study and if things are done the way they're supposed to be or you intend to do. And in addition to that, the studies are usually monitored by Data Safety Monitoring Committee. Some of them are institutional, some of them are external, looking into toxicities that might happen when patients are admitted to hospital and they are a clinical trial participant, really looking into why were they admitted, was that related to clinical trial, for example, and also are the clinical trials being, accruing participant at the rate that is feasible for the study conclusion. These things are all monitored on an ongoing basis. So we talked about what clinical trials are and why they matter, a little bit extensively about clinical translational studies and phase 1 earlier, but all clinical trials, when you're participating, they start with informed consent. And I think in this section about partnering with you, I think it's extremely important that you understand informed consent is the important part, keyword is informed. I oftentimes hear in clinic, Dr. Umemura, I want to participate in clinical trial. I don't care what it is. As long as you think it's good for me, I don't care, just tell me where to sign. And we have to always review. It is very important that participant understand the importance of why the clinical trial is being done, why that particular trial is talked about, why we think it's beneficial to that person in the community, all those things. It's very important for the physician who is consenting the patient to really have that conversation so you can understand, participants understand, because essentially the participation and withdrawal from trial needs to be voluntary. There are different kinds of clinical trials. Some are blinded, meaning so single blinded cases, the participant may not know if they're taking what kind of medication, for example, or if it's double blinded, placebo and the therapeutic drug may look exactly the same, and physician and investigator and the participant, neither of them would know what kind you're taking. There are randomization in trials. So for example, if they are clinical trial arms comparing normal standard of care therapy to the investigational therapeutics, you may be randomized, usually by computer, in terms of which arm that you will be on to eliminate bias, selection bias, or it may just be one arm non-randomized. And sometimes clinical trials would use crossover design, where you will start in one arm and at certain particular points, like progression of disease, you would cross over to another arm. And these are all different kinds of design you may see. Clinicaltrial.gov was mentioned earlier. It is a place where all clinical trials are registered. There are kind of high-level information that is on there, not every little detail, but usually eligibility criteria is included. So that's usually where I would go when a patient brings up a clinical trial that I don't particularly have. But in other centers, we try to look through to see if that patient may be eligible. I can also find a contact information to reach out to, for example. So why do we need to do clinical trials? We need clinical trials not only to find a better treatment for brain tumor, but also to have a better understanding. I think it's same in everything in life. You can't really improve things unless you really understand what's going on. Same thing with in medicine and brain tumor in neuro-oncology. You also need to compare if one thing is better than the other. And also earlier we talked about we heard about translational therapy, where preclinical basic science studies can be translated into clinical studies. And these things are all studied in clinical trials. I won't spend too much time here because I think we already heard about how clinical trials are started in the last two sessions. But essentially you have an idea and you have something to back up your idea. And then you come up with a question, hypothesis, and a means to measure if your hypothesis can be supported, which would be the result or endpoint. And once those are the fundamental to create a protocol for clinical trial. And protocol would be the place where you state how the clinical trial will be conducted, including eligibility and how things are measured and how statistical analysis will be done. And you have to set these things ahead of time. Now, preclinical studies are the basic science studies that are backbone for the clinical studies to be done. And we heard about that earlier in the first session. And once the preclinical evidence is there, let's say, there are evidence that certain medication may work in animals, we need to see if it is safe or tolerable in humans. And usually we don't jump straight from ideas to humans. There's usually some sort, I think in Q&A, there was a question about if there are toxicities that you can anticipate. Usually we have an idea of what kind of toxicities we will anticipate in clinical trials, and those things are usually in part of informed consent. The phase 1 studies, so I'm going to touch upon what phase 1, 2, 3s are in kind of basic idea of what they are. A simplified way of thinking phase 1 is it's kind of an entry point of how much can we give? Is it safe? Is it tolerable? And how often to give? Those are usually the main question of phase 1 studies. And because the questions of how much, how often can be answered in smaller population of patients, the phase 1 studies are usually smaller in size, about 20 to 30 patients in size. And sometimes that can be combined with phase 2 studies. Now, phase 2 studies usually focuses on, does it even work? Like, let's say it is safe and to be given to humans, does it work? And that can be combined with phase 1, that can be combined with phase 3. And usually those is it effective is measured in different ways. One way you can measure is if the size of tumor on MRI is the same, if it shrinks, if it gets bigger, or the time from when you start the study to when that happens, so time to progression, or overall survival of particular participant and in overall study. So those are all different ways to measure effect, and you the investigators need to define what that primary outcome would be for a study ahead of time. And there may be other secondary questions in phase 2 studies. Phase 2 studies are usually bigger than phase 1. It usually involves somewhat less than 100 patients. It does take a couple years at the least usually to complete accruing patients to the study. But those duration of the studies really vary by what kind of tumor it is, how rare the disease is, and how many sites the studies are open at. And phase 3 studies, I think a lot of people think of the phase 3 study as the most exciting study. A lot of times I hear patients say, is this phase 3? Has this been tested at phase 3? I think it's the common one you hear. So phase 3 studies are usually studies that ask if particular treatment is better than the other one. And that is commonly done in randomized method where you have one type of treatment compared to another type of treatment, and you're randomly assigned to one or the other. Sometimes they are blinded, and you usually have a controlled arm. You will then, if successful, if you can show that it is better, you can apply for FDA approval for specific use of that drug for that particular use purpose. And phase 3 studies usually require several hundred patients for that statistical analysis. And there are other therapeutic trials that are combined to phase 1, 2, 3. We have already heard about phase 0 window of opportunity study, which is extremely important. As an example, we have a clinical trial at University of Michigan that looks into, there's an old drug that may have benefit in glioblastoma treatment that was being used for various other diseases that has nothing to do with brain tumors. Now we want to know if it actually gets into the brain, if it is actually doing what we hypothesize that we see in animal, if it's happening in the humans. So we have a window of opportunity study where a subset of patient in our phase 1 study, a subset of patient in that study would then take medication for 1 week prior to their planned surgery. So we can answer those questions and send those tissues back to the lab so we can better design future studies. There are also expanded access program, which is kind of in between trials and compassionate care program, I think is the easy way to think about it. There may be drugs that have some hype or some promise in treating brain tumor, for example. And if it is FDA approved medication, sometimes you may hear about compassionate care program or free drug program where you can apply to the company to give you the medication if it's not covered by insurance. But if it is not an FDA approved compound or drug, you can't really go that route because they can't give it to you. So usually there is a clinicaltrial.gov FDA registration where you have to apply to use this particular drug through IRB, through FDA, so we can get that drug to a particular participant. And then sometimes that is just one person IRB application, one person FDA, and that's called expanded access program. Phase 4 study is usually the post-marketing on a large scale study to see if there's anything that is noted after it's been approved. Now, I'm going to address a couple of things. I also have an FAQ slide at the end, but I wanted to do a little bit of myth buster today in my portion of the talk. So sometimes I hear about, I read this on the internet. This came up on New York Times. What do you think? Like, why do you have to do a study? Why can't you just give it to me? So, if someone were to ask me, do all the alternative approaches, do all the non-standard approaches, do they all have to be studied? Why can't you just do it? The short answer is yes. You have to really study them because sometimes you hear, oh, there's this anecdotal evidence that this particular treatment worked for so-and-so. It's not really an evidence where we can apply to a large population. Unless we do a study, we we don't have a prospective data in terms, that eliminates bias of retrospective data. And we need to really, like I said earlier, we really need to understand how things work to really apply to future studies. And we really need to understand if we use a particular treatment, if we can anticipate the same type of effect in large population, or what kind of population can we apply that to? And then our biggest concern would be, just because it was fine for one person doesn't mean it is fine for everybody else. And we really need to eliminate harm. And all these things needs to be considered and needs to be done in a research study. So who conducts clinical trial and how are they conducted? Clinical trials can be conducted by independent researcher, independent institution. Sometimes we all get together. Many of us are in clinical trial consortium together where we run national studies. National institutions like NIH also would run studies. And we all know pharmaceutical companies also run clinical trials. Within each trials, you always have a principal investigator who is essentially in charge of the study. But the principal investigator cannot run a trial by him or herself. You'll usually have co-investigator, other physicians, and also clinical trials are supported by myriad of staffs that make it possible, including nursing, including people who enter data, including people who dispense the medication, and statisticians. So it takes really a village to complete each clinical trials. We've heard over and over today that you can't separate money from clinical trials. Unfortunately, the money does matter. And clinical trials are usually funded by either government grants, foundation philanthropy funds or donations, companies or institutional funds. And when it's time for you to consider if you should be in a clinical trial, like Larry mentioned, the biggest first thing is, are you eligible? There may be different types of eligibility considered, like where are you in the diagnosis? Are you newly diagnosed? Are you at a recurrent disease? What kind of molecular markers does your tumor have? How functional are you? Are you able to walk? Are you able to swallow pills? There are many eligibility criteria to consider. And we really also need to understand what it really means to you and then the cost it would be to you, not just toxicity and not just the money, but can you travel to certain places? And what does that mean to you in your life? Taking time away from your family. And these are all part of cost, money or not money. Is the trial available at the center you're being treated? These are all the things to consider. And I'm going to skip this slide for the time, and also because I think it's being covered by today's talk, and just talk about a few of the common things that come up. So when someone says we need to do clinical trial, but this drug has been FDA approved for other reasons, FDA approval is not just about if the drug is approved as a blank state approval. A particular medication or treatment needs to be approved for a certain indication. Just because a medication is approved for colon cancer doesn't mean it's approved for glioblastoma use. So FDA approval means a couple different things. I think there was a question about placebo earlier in the Q&A when I was looking at the Q&As. Am I going to get placebo? In brain tumor treatment, you're not going to come across a clinical trial where you're going to be only getting placebo when the standard of care is not observation. So those are one of the things that come up. The placebo is oftentimes given in addition to standard of care if we're comparing to another trial, because placebo does give some effect. Placebo effects are real effect. That's why we do have to study in comparison to placebo. Clinical trial participants sometimes says, has this worked in others? Is it working? When am I going to know the result? I'm being on this study for a year. And these things are reported on an ongoing basis at scientific meetings, but then the journal papers usually don't come out for about a year or two after the trial participants are finished on accruing to the study. So these are not something that your doctor may know the answers to, but may be able to find out on an ongoing basis. And I hope I was able to cover the basics of clinical trials, and I'll be happy to answer any other questions later in the Q&A section. Thank you.
All right, Susan, we're going to hand it over to you. Dr. Chen?
Yes, thank you so much. Are you able to hear me?
We can hear you. I'm
trying to.
There you go.
There we go.
All right, we're going to give remote control to you.
Thank you very much.
First
of all, I wanted to thank Emily and also the ABTA, of course, and Mike, as well as Yoshi, for this incredible sessions today. Really important about how we partner with you to advance research. So as you've heard, the sort of getting to the point of doing clinical research is so much that goes on, but we would never be able to understand the benefit to you unless you all helped us. And I think that is the main message that we want to give to you today, certainly with Yoshi and myself in this particular session. So these are my disclosures. So I think the first thing is keeping the patient at the center of everything we do, which is so critical. We have this multidimensional team approach to foster research. And we can understand both the basic science information, as well as using animal models and computational biology, using multiple databases. And then all of the clinical aspects of taking care of you is so critical as well. And this is where the clinical research versus medical research involving people. So you heard from Yoshi very elegantly about how this all comes about. And so the spectrum of clinical research, I want to emphasize, is more than just therapeutic advances. Of course, we all want to understand and learn what's the best treatment for our patients. What can we do to improve the survival of patients? But we also want to understand ways to detect it, to control it. And then what about quality of life? So those are all still problems and things that we need to address when we think about patient care. And so when you think about how this whole process starts, we have the identified problem. There's a lot of supporting information to address the problem. And then we come up with the methods and testing. This is where the clinical research piece comes in. So patients play a critical role in research. You can volunteer to be in a study. So you've heard about being in a therapeutic study, but you can also give permission to have your medical records reviewed for research purposes. And you can also agree to donate blood or tissue samples that could be collected and further studied. I think you've already heard too that when a study is offered, it's really up to you to choose whether to participate or not. And if you choose to participate, you can certainly leave the study at any time. So I think that those key principles of partnering with you with advancing the types of information about how best to take care of patients is really important. So just to think about non-therapeutic clinical research studies, these can span so many different types of ways to understand the epidemiology. In other words, what are some of the environmental factors that may cause cancer? And then there's genetic factors. So what is it about genetically who we are that might, in the combination with environmental factors, influence the risk of either developing a disease or actually having the disease progress? There are also many natural history or registry studies where we can follow a group of people over time to collect health information to understand how the disease progresses and what the long-term effects of treatment might be. There's screening trials to understand diseases at their earliest stages So understanding, how do we, if a patient presents with a symptom, for example, how do we know that this is representative of a diagnosis of a tumor? And then how do we diagnose progression so that we can intervene as quickly as possible to control the disease? There are also diagnostic studies or tests or procedures to understand the condition. There are quality of life studies to explore and measure ways to improve how patients are doing. And these include non-therapeutic intervention trials that can test new therapies to enhance care. There are also medical record and database studies that review information. So this is collected already, and we can also go back and look at that information to understand treatment and outcomes. So I think to make this real for you, I just wanted to talk about some clear examples of how this can be done. A lot of these are ones that I've used from UCSF, but certainly there are many, many institutions that are doing this type of important work. So when we talk about genetic and epidemiology studies, this can often involve surveys about your general health, as well as environmental exposures. For example, what kind of work did you do? Were you exposed in your work to other carcinogens, for example? There's usually a control group of individuals needed to assess the risk, and people are often asked to give blood or sometimes a smear of their mouth, because that produces some cells that could be tested, or the fluids to allow part of their tumor to be tested as well. And this is really important to understand the link of genetics and how tumors form and progression. So this is an example of one that is being conducted at UCSF by my colleague, Jenny Taylor. And this is taking firefighters who are known to have an increased risk of cancer, not only brain tumors, but bladder cancer, lung cancers. And this is because of the exposure to byproducts of fires that might contribute to the development of the tumors. And the data suggests that this level of this particular compound, haloalkenes, may be a marker of risk. And so we're studying firefighters in California. Of course, unfortunately, we've had large fires in the last few years. And the plan is to acquire blood samples and to track the risk of developing a brain tumor in a large cohort of firefighters in California. So that's just an example. Another one is one that my colleagues, both at UCSF and at the Mayo Clinic, published on an example of how we look at genetic variants. So these are things that patients, their normal genetic makeup, that can be associated with increased risk of glioma. So we understand that there's both what you come in as a patient, but also what are acquired that may trigger a tumor to be formed. And these two things were included and really correlates to certain subtypes of brain tumors and glioma. Now, natural history or registry studies are also important. This is an example of a molecular genetic host-derived and clinical determinants of long-term survival in glioblastoma. And this is the clinicaltrials.gov number. So these are patients who have a history of glioblastoma who survived more than 5 years. And these are what we would call the outliers, right? I mean, when you think of glioblastoma and the statistics about the expectation for how patients do, we know that not every patient does the same. And in fact, we have a number of patients who survive more than 5 years. And so we are interested in understanding what are the patient characteristics? What kind of treatment do these patients have? Is there something about the tumor that might be important? How are these patients doing at 5 years? What is their cognitive function and quality of life? And how are their caregivers handling this have the journey affected them? And so this is a study that is an international study that is looking at all of these dimensions of a patient and their lives. And this is one that is supported by the Brain Tumor Funders Collaborative, for which the ABTA is a contributor. So using this type of multi-institutional, multi-international studies, we can get at this cohort of patients who are surviving for a long time with glioblastoma. Diagnostic studies are usually imaging studies we think about, because most patients, as soon as they have a history of a seizure or a headache, oftentimes an image is obtained in the brain. MRI scans are standardly what I'm sure a lot of the patients here on this call are all familiar with MRI scans. And one of the key challenges with MRI scans is it's very good at telling us anatomy, but sometimes it's very confusing what it is we're seeing on the scan, especially after the treatment. And so it can be very difficult to say, is this because of the treatment I've had with radiation, or is this because the tumor is actually growing? And so we are always looking at better ways and procedures to diagnose a condition. And this is an example of using the fact that tumors have different metabolism than normal brain. And so we can image the metabolic aspects of things, then that might tell us about ways of which, we can differentiate between tumor and normal tissue. Screening studies are ways to identify diseases at their earliest stages, either at first diagnosis or at progression. And this is an example that's being conducted by another colleague, Jenny Taylor and Jenny Clark, as well as Dr. Molinaro, looking at evaluating novel blood biomarkers of progression and clinical outcome in all patients with glioblastoma. And so these are patients that we capture before surgery and get a blood sample. We then do surgery and follow these blood samples over time. And what's interesting is that by collecting these samples, sometimes it's important because sometimes we don't know what the right biomarker might be. But what we have then is this amazing collection of samples all the way through the trajectory of the illness of that patient. And then we can go back and test as new technology and new ways of evaluating genetics and tumor cells in the blood, for example. As we go forward with understanding that, we can now go back and get all of that amazing tissue and samples and clinical history that's been collected to do that. Now, quality of life studies, I think, are really key. So as we've talked about, it's not only about prolonging life, which is what we are hoping to do when we have new therapies, but it's also making sure that the quality of life is maintained or improved. So these can include caregiver and health provider assessments, and usually involves what we call patient-reported outcomes. So it's you telling us how you're doing. And that's important. So there are many international studies using surveys to assess quality of life for patients and caregivers. Some of these quality of life studies can also incorporate the use of treatments. In other words, here's an example. If patients are having a lot of seizures, we may want to test a new seizure medication. And we do that in the setting of a clinical trial. The endpoint of that is not whether the patient lives longer, it's whether the seizure is controlled. And so you can see and what their side effects might be and what the quality of life is. So you can see that quality of life studies can span not only understanding what it is that the patients are experiencing, but also ways to improve. Intervention trials that are not tumor-directed, so we can test new therapies to improve care. And this is an example for, they talked about seizures. This is another one of looking at the feasibility of cognitive rehabilitation strategies in stable patients with lower grade glioma, which is being conducted at UCSF. And this is where we are being very proactive of saying, if we anticipate that patients are going to have cognitive changes, are there things that we can do to decrease the risk of that happening? And so these cognitive rehabilitation strategies are ones that we're exploring. We can also use medical records and databases, and this is a review of a large group of patients. This is a cohort of over 1,000 patients with newly diagnosed glioblastoma to examine the effect of the extent of surgical intervention on survival. None of this work could be done unless we pool all our information together. So this is, again, another multi-international study that took patients with glioblastoma, information about the extent of resection, information about their clinical outcome, and putting it all together to understand the role of surgery, for example, in affecting the lives of our patients. So in summary, I just wanted to share with you, there's so many partnership opportunities for patients to be involved in clinical research. And although testing new therapies is critical to advance the field, these other types of clinical studies provide really key knowledge about the tumor development, the diagnosis, the monitoring, as well as side effects of the disease and the treatment that can affect quality of life. And so, you play a critical role in the research process overall. And we're just so glad that we're able to be here to share all of this with you. So thank you very much. I want to acknowledge Naomi Hoffer, who is the program manager of the Sherry Sobrado-Brisson Brain Cancer Survivorship Program, a key partner with me, who helped me with the slides. And I just wanted to, again, thank you all for the opportunity.
All right, thank you both so much for the wonderful presentation, and thank you to Larry for sharing more about his experience earlier on. We now have time for questions. As a reminder, if you have questions that you'd like to ask any of our presenters, please type and submit it using the Q&A button on the bottom of your screen, and we'll answer questions as time allows. All right, so let's see what we have. All right, so one of the questions that we have here is asking, Dr. Chang, you shared some great ways that patients can engage in clinical research, like volunteering in a study or having their medical records reviewed. How can patients find these opportunities if they aren't available at their specific treatment center or if they aren't brought up to them by their healthcare team?
Thank you, Ambreen. That's a really great question. So actually, clinicaltrials.gov does have non-therapeutic studies as well. So some of these may be available. Certainly all of our imaging studies that we do, for example, are available. I often actually get questions from patients who call in and say, I saw this study. Sometimes I don't quite understand it's not a therapeutic study. So it is available there. And I think it's just, again, looking at the ABTA website sometimes can also have information about registries and other opportunities for participation.
Thank you so much for sharing that. And also you talked about, the multidimensional treatment approach to care when it comes to trials and research. Who then becomes the point of contact for the patient if the patient has questions about side effects or how they're feeling? Who are they reaching out to when there's so many members on the care team?
Yes, so it can be very confusing because a lot of times, as you know, patients are usually very well and then they get a symptom and then all of a sudden they have to deal with the diagnosis. So I would say it depends on the therapy. So when you're hospitalized and you're having surgery, obviously any effects from the surgery itself, you need to contact the surgical team, your neurosurgeon and their assistants and so forth. So making sure that you understand before you get discharged, what some of the things to look out for. Mainly it's things like, headaches, seizures, wound issues. So that would be your neurosurgeon. And many times after you're seen by the neurosurgeon and you have a diagnosis, you're often tapped into a neuro-oncologist. And I would say I see the neuro-oncologist almost like a quarterback to help with guiding, not only talking about what the disease is, what to expect, but who also is going to be part of your team. Are you going to need radiation therapy? Is there a clinical trial that you have an opportunity for? I think Yoshi talked about that really elegantly about how talking about the different options is so important. And so it depends. So if you're in a clinical trial, the most important thing is that it's your team that has allowed you to be consented. That's the team that you need to let know about any side effects you might be having. So checking in with them is very critical. Obviously, if you're undergoing radiation therapy, then it's your radiation oncologist because there could be skin changes and other things related to the radiation itself. But we work so closely together as a team. And I think that is the most important thing, is that communicating, if the radiation oncologist notices that there is something that's happening that's not the usual, it's a quick phone call. Dr. Chang, could you see that patient at the same time they're coming in for radiation, to check in on them? So it's really this team effect that's important. But your neuro-oncologist, I would say, is always somebody who can help you navigate the whole situation.
Thank you so much for sharing that. And Larry, I know in your experience, you had a very good experience with your treatment team who was supporting you through your clinical trial. Can you share a little more about what the communication looked like with your treatment team and what benefits you felt like you got from being on a clinical trial?
Sure. The team in itself, the trial coordinator is so key. And we could go to her at any point in time, and she had communication with the rest of the team. So we didn't have to sort out and try to run someone down, because all the doctors are busy. But we also always had the cell phone numbers of the key team members. Dr. Forsythe, I could call him at 3 a.m. in the morning. Fortunately, I didn't have to. But The communication process was made very clear. It was encouraged. And also the point that one of the doctors was making is the communication with the doctor. Any issue you have, something changes, something feels differently. Get that information out quickly, because even in what was in the 48-page consent document, when I actually had an adverse event with ALT-AST, but I knew from the document exactly what treatment was going to be addressed to me before it occurred. So the communication process from the document to the team was really totally connected. And we never felt, me and my wife, of course, she's a key part of this, just not time to go into all of that, but we never felt unconnected, and we knew that we could get an immediate response if something occurred.
Great. Thank you so much for sharing that experience and what it was like communicating with your team. We have a question here about seizure management. Dr. Chain, you talked about the importance of quality of life, and seizure management can be a big part of that. Are either of you, Dr. Chang or Dr. Mimara, aware of any studies that are being done on new or better ways to manage seizures?
I can take that one. So seizures is one of the big component of what we do. But I think you've kind of seen the collaboration between disciplines today. We have neurosurgeons, we have basic scientists, neuropathologists, collaboration with patient neuro-oncologists. Epilepsy is its own subfield in neurology. And although a lot of neuro-oncologists are neurologists and we do manage seizures, I think the Q&A that I'm seeing the person is on 3 medications. I think if you have uncontrolled seizure, refractory seizure, it is important that you see maybe an epileptologist who can partner with your oncology team so you can get the best care possible, especially if it is a complicated seizure management. There are
sometimes I was just going to add that surgery, when you talk about epilepsy team and the like, sometimes surgery can contribute to that. And also one other area that we haven't really talked about a lot is radiation therapy. We know that radiation therapy can, when it's given, actually help reduce seizures and low-grade gliomas. I actually just did an anterior temporal lobectomy on a patient who had a very extensive tumor, but was having temporal lobe seizures, and it actually helped to reduce the seizure frequency. So we weren't going to cure his tumor with the surgery, but we knew that we was going to make his life better as a result of doing that.
And also, the tumor itself can, like the mass can cause seizure, but also, low-grade gliomas with IDH mutation. IDH mutation doesn't just cause cancer, it can also be a causative component for seizures because of the metabolite. There are actually some epileptologists, for example, Dr. Avila, MS Memorial Sloan Kettering, there are epileptologists who specifically study brain tumor related seizures. There are clinical trials, like there's a phase 3 clinical trial, the acronym is INDIGO, but for low-grade gliomas, observation versus IDH inhibitor. In that study, seizure, how often people are having seizure, and that is also one of the things that are being collected within the study as quality of life.
Thank you so much for sharing that insight. Another question we have here is Dr. Mimara, you touched on expanded access briefly. Can you maybe explain a little more of what expanded access means and how patients can gain access to more treatments using this, how they can apply for treatments through expanded access?
Sure. So I think it can be quite confusing. People talk about right to try law and expanded access and compassionate care. There are so many different ways one can get medication. I think the first time I gave IDH inhibitor, I know this is recorded, so I don't know if this is kosher, but I just wrote for it because I knew it was an FDA approved medication. I wanted to give it to my patient. And so sometimes you can get FDA approved medication for non-FDA approved indication by either prescribing it and insurance covering it, or companies giving it to you by free drug. But if it is not an FDA approved medication for any indication, you really can't do that. So that's where expanded access comes in. So usually there is a clinical trial protocol, just like a clinical trial. You still have to do IRB approval. You still have to have an FDA approval. You still have to have an informed consent. And usually there are data being collected about toxicity, survival, different things, imaging characteristics, just not as stringent as traditional clinical trials. Patients can get into that usually by physician or patient reaching out to the company that is making the medication. And sometimes there is a portal that you can submit requests to. University of Michigan, we have a group that handles that. And sometimes we do a single patient application, or if it is something that we are using for multiple patients, we might have a multi-patient application. It unfortunately doesn't guarantee that you can get it, because just like clinical trial, there are eligibility criteria. And also some expanded access program may not be available at every center. So I think the best thing is talking with your physician. Sometimes your physician can talk to contact at the company, and they may have recommendation of where you can go if it's not going to be available at that certain institution.
Thank you so much for that great insight and advice for our audience. That is all the time that we have for questions for this session. I will go ahead and turn it back to Emily, and we'll move forward with the rest of the program. Thank you all.
Thank you so much, Larry and Dr. Iwamura and Dr. Chang, for sharing with us your experience and expertise with clinical trials. I know we will see you briefly for our next session. So right now we do have a short 5-minute break, and then we will finish out our last session of the day, all about challenges, overcoming barriers to progress, where it will be a didactic panel discussion. Thank you so much, and we'll see you back in just a few minutes. Welcome back, everybody. I hope each of you took a brief moment for a break. I'm going to hand it back over to our chair, Dr. Vogelbaum, to introduce our very last session of the day and introduce our panel speakers.
Thank you, Emily. So in our last session for today, titled Challenges, Overcoming Barriers to Progress, we're going to have a panel discussion to explore the challenges that impact the development of cures for patients with brain tumors. I mean, so far, you've heard about all of the research that's being done at many different levels to try to more effectively treat and cure brain tumors, but we haven't done that yet. Why not? I welcome back our former speakers, Larry Thomason, Dr. Sinai, Dr. Heimberger, Dr. Mimura, Dr. Chang, to explore some of the issues and some of the barriers that we've had to reaching a cure. So I'm going to start with some questions about the funding and costs of doing clinical trials. Amy, you talked quite a bit about how much it costs to get a therapy from the laboratory finally to patients, the millions of dollars that you documented very effectively, but there are other costs to running these clinical trials, not only for launching them, but also on the patient side. And Larry, I don't know if you're on camera now, perfect. But I was wondering if Larry and then Suzanne, if you could talk about the patient side of the costs. You know, what it's like. You know, Larry, you live 6 hours away from Tampa. What was it like having to travel frequently to Tampa? What impact did that cost have on you and your wife? And you're muted still, Larry. Larry, you're muted.
Okay, can you hear me?
Now we can hear you. Thank you.
Yeah, we took a different approach because while I was asymptomatic, Dr. Forsythe fully expected I would run into seizures quickly. So what we did, we were fortunate to be able to obtain a rental unit. So we just relocated to Tampa from where we were and stayed for a couple of months. And obviously wasn't without cost to do that, but it was much more convenient And when you have to drive 6 hours, there's one way, there's cost associated with that. So on the, I won't say on the advice, but the encouragement of Dr. Forsythe, we relocated so that if we had unusual events occur, we would be in a position to address those quickly. So obviously, if you're an hour away, you'd probably take a different approach. But fortunately, we were able to relocate and handle things as more of on a local basis than other than the cost. It was like we lived there, which we, for all practical purposes, did during the first stages. And I'll also add it, I'm not sure I'm a good representative of someone on a clinical trial, because after 5 weeks, 65% of all of I actually had some in the lungs too, but after 5 weeks, 65% of my issues had dissipated. So we were in a complete stage of encouragement after 5 or 6 weeks. So it wasn't like we were dealing over a period of year with a totally stressful situation. We quickly moved to an optimistic situation.
That's wonderful. Thank you, Larry. Suzanne, do you want to talk about some of the efforts to support patients who need to travel for clinical trials?
Yeah, I think this is such an important question because it gets at, what is it like for a patient and family to participate in a clinical trial? And so, in addition to the fact that just the whole process is so complicated, many times with clinical trials, the testing and the assessments are much more frequent than standard of care. And especially for very early studies where we're trying to understand like how much of the drug is getting into the blood, what does it look like, how does it clear in the bloodstream, are we getting enough of it in the blood? You've already heard about the window of opportunity studies where that mandates the patients have surgery. That's usually part of their care, but it does interject more intensive types of assessments, which sometimes might require travel. And so when patients live far away, we worry about access to care and the equity of who's participating in these trials. The other thing to remember is that many of our patients, for example, may not be able to drive. They may not be able to understand all the nuances of everything they need to be doing, the timing of all the different tests. So many times caregivers are needed to help them. So that means the caregiver has to take the time as well, take time off work and to travel with their loved one to get these studies done. If there are side effects from these treatments, then that again causes potentially more interventions for them. And so I think supporting the patient in terms of letting them know what to expect and the family as much as possible in terms of how many visits that are needed and making sure that that is minimized. And I think that's on us as well as clinical trialists to make sure that we're minimizing the types of number of blood tests that we're asking patients to come back for and scanning and things that are impactful in terms of sort of the cost to the patient. So I think that there are many ways to try to alleviate those types of expectations. And also, I think building in, for example, in the phase 1 studies with companies, for example, to make sure that any costs for overnight stay is covered, for example, for the patient and their loved ones.
So I'm going to shift gears to another barrier that we often think of when we're trying to run clinical trials. And Amy, you showed a great image of a huge stack of documents that had to be prepared to get an IND. And having been there and done that, I think as others in this session have done as well, one might say, is the FDA getting in the way or not? What are your thoughts, in terms of regulatory barriers to getting new therapies up and going for brain tumor patients?
Well, I don't think they're going to grant an exemption just because we're in a rare cancer. So I think we have to be a little bit realistic here about trying to change the FDA and how they do their business, so to speak. You know, I'm like, I don't have a good answer for how the FDA can do better on this, other than put more people on it, approve it faster. But usually they do it within 30 days. It really does come down to the rigor that's necessary. You know, you don't want this done in some secondhand lab and make a mistake with regards to, you know, any of these kinds of important criteria for safety for patients. So I just don't see it going anywhere, unfortunately. And that's just the burden of actually doing business. It does squeeze out some of the small pharma companies, because a lot of times they have to go through Series A, Series B to get enough venture capital to actually get these kinds of studies funded. And typically they're done through, you know, contract research organizations or CROs. A lot of these are actually located in China or India, so they are cutting some cost, but still it has to be in a certain accredited labs, et cetera, to make sure that it's done rigorously and with appropriate oversight when you do these kinds of studies. You know, you just don't want to do it in some, you know, sort of, you know, backwoods trailer, you know, with a couple of, you know, stray dogs you find off the street. So, you know, it is the process that we have in place.
Yeah, you know, I think that you know, people may not be aware of some of the disasters that occurred before there was a regulatory apparatus that was effective in place. Things like thalidomide being used for to control morning, you know, morning sickness, you know, and that could have been avoided. And then there are examples with devices as well, metal and metal hips, where there was not an effective reporting mechanism when there were all sorts of problems going on with those. So it is a necessary part of this. And one might also argue that sometimes the FDA has bent over backwards to help us with some approvals in our space that were based upon clinical trials that would not have led to an approval for lung cancer or for breast cancer or for melanoma, because it wasn't as strong data, wasn't as many trials as they normally would require. So it is a balancing act. Unfortunately, as you said, it is a real part of the system. It's a real cost, and so we have to work with them, and we do a lot of that. Nader, I don't know how much you've been working with the FDA on some of your trials, but you know, you see it on the device side as well.
Yeah, no question. I mean, it's a necessary mechanism for sure, and I think we all want things to move faster, but you know, we don't want people to get hurt. I think there were some questions earlier in the seminar about toxicity, and you know, there are no new therapies that don't have side effects. They all have them, and it's up to us to make sure that it's not the side effect that really stops therapy for patients with difficult diseases. So we're all trying our best to navigate those mechanisms.
So one of the questions that came up earlier related to the fact that you know, one of the attendees said that they had never heard about certain of these trials from their caregivers, and Yoshi, I wonder if you can take on that topic of raising awareness of clinical trials, especially for people out in the community. You know, when their local physicians may not be as aware as we would like, and how do patients become more proactive and learn about these trials? What have you all been doing at Michigan to help with that?
So I think there's so many levels, you know, so I think it's important for me to be aware of what everyone is doing. So I try to pay attention at the national meetings I go to. There's different challenge to that for academic physician to be able to go to these meetings. Having the Zoom meetings certainly cut the cost, but that's a different challenge. But we can't know everything. So what we try to do is, if patients want to know kind of second opinion, we try to help them get a second opinion at big centers like Memorial Sloan Kettering, MD Anderson, UCSF. And when they bring back information, I try to think it through with them by looking at the information they bring back. So I'm kind of their at home information Google, for so to speak. And another thing is in our after visit summary, we in Epic, we have smart phrases where we do put down the ABTA website and Brain Tumor Support Network because they do have National Navigator. They are philanthropy funded, and it's wonderful that they have navigators that look into what clinical trials are available and they try to match you up. I know that they are quite thorough because every time I update anything about my clinical trial at clinicaltrial.gov, I get an email from them asking about eligibility. So I think those are very good resources. And additionally, I think patients do help each other. There are patient support groups that, pandemic, there are a lot of obviously downside, but some of the upside is everyone is tech savvy now. And people from California and New York and Michigan are connecting on social media support groups. I have a patient who have quite a standing on social media who have raised a lot of awareness by writing a book, by having a TED Talk. I think patients also very much help each other as well. And I do want to add, I do want to add one thing. I realized we didn't necessarily touch upon equality. I think a huge part is, right now with everything we're doing, we're still not really breaking into patients who have a lot of resources, have computers, have the smartphones, obviously they get more information. People who don't have resources, they really don't hear about a lot of options. Sometimes they won't even think to see a neuro-oncologist until there's really not much I can do. And I think community research, those are the areas I think there's still a lot of shortages that we need to really work hard on. I speak with my patients. Sometimes I ask my patient, someone is African-American, I say, what can we do in your community? And they often say, have a talk at a church, something like that. So I feel like we're such a fundamental level, nothing high level, but I think there's so much that we have to do in terms of awareness.
Yeah, I'm glad you brought that up. One of my colleagues has just put together a group to start doing exactly that at our institution, because we know of communities within our region that are not getting access, and we need to help them by going into their communities and help them understand what opportunities are available. Suzanne, I think you were going to make a point.
Yeah, so in addition to education and sharing information among patients, which, as Yoshi pointed out, the ABTA does such a great job at, I think it's a responsibility for us as people, working in an academic institution where we collaborate with our community oncology partners. And it's really for us to also engage with them so that they are aware that clinical trials are available. And so there's so many networks now that are happening, right? So most hospitals don't just work by themselves anymore. There's all of this collaboration across different areas, and reaching out to our colleagues as the experts and as neuro-oncologists and neurosurgeons and radiation oncologists, et cetera, who take care of brain tumor patients, because most of the community oncologists may see maybe 2 or 3 in a year that they see with a glioblastoma, for example. And so it's having that relationship built out on a professional level that's also going to help the patients in those communities as well. So I think it's going to be both at the provider level for us, but also at a patient level. Thanks.
Larry, the point of patient support networks and the like, how much have you been involved with them, and is that how you help support others in your position?
Yes, I had given Dr. Forsythe and his team, anybody, they have my phone number, they have my anything, email or whatever, and if he feels that someone could benefit from my experience, he texts me and says, or emails me that he'd like this person to telephone or whatever, and he has carte blanche. He doesn't even have to do that. Also, what has to do, I probably talked to maybe 25 different patients or potential patients or people considering a clinical trial or people who are having issues. So me and my wife both are participating in that. but it's through his direction that that's occurred.
So, Amy, what do you all do to try to recruit patients? Because we know that one of the challenges that we have in running clinical trials, and this is one of the internal institutional challenges, is that if we open a clinical trial and we don't get patients on that trial, our institution closes it. And the reason is that there's a carrying cost. Having that trial open incurs a certain number of costs, and ultimately the goal is to treat patients. And if we're not doing that, then we're not justifying that cost. So we need to recruit to those trials. What do you do, Amy, to make sure that you're moving things along with your trials?
So there's two comments I want to make. The first is it's fully recognized that there's insufficient community enrollment on these kinds of clinical trials at the very highest level, including the National Institute of Health. And I think moving forward in the next year or two, we will be seeing either some kind of supplement put forth through the Cancer Center grants, et cetera, to increase the diversification of the kinds of patients we put on clinical trials. But to specifically address Michael's comment here at Northwestern, it's not just the mothership. We actually have 13 affiliated practices, community practices that surround the Chicago area. And we actually do, and I suspect there's other folks on this phone call as well, where we do multidisciplinary tumor boards that incorporate our community practices. And so we have our neuro-oncologists out in the West region, in the North region, the South, and we mandate as much as we possibly can clinical trials opening at those affiliated centers. What works very well for those community centers are, some of the alliance clinical trials, some of the things that do not involve PK, i.e. phase 1 clinical trials, because a lot of times you need a lot of frequent blood draws, and really the community practices are not really set up for that. But there's certainly many of the phase 2 clinical trials that we open and have actually, in some cases, even better enrollment out there than we do at the mothership, so to speak. Especially our radiation protocols do very, very well out in the community practice of photon versus proton kind of studies. I think we're going to see more and more of these network initiatives where we embrace our community partners. And I see Susan's nodding her head. I suspect she probably has some experiences at UCSF as well with this kind of thing.
Nader, how about in Phoenix? You're muted.
Sorry, yeah, I would echo everything Amy said. It takes a village. And a common question we've gotten on this seminar is, how do we find out about these trials? How do we access them? Am I qualified? My local oncologist hasn't identified for me these types of experimental options. So to some extent, it's incumbent upon the patient and caregiver population to be proactive, to get all the other opinions and to seek out trials. We tend to field many patients who are self-directed because of the connected world we live in. But I think it's important as the patient population to remember that we're all on the same team here. And these community physicians that you may be seeing, while they may not have direct knowledge of all of the trials available, they're not going to stand in your way. And in fact, they're going to want you to get as many options as possible. So partner with them, get other opinions, use all the resources that we see. And I think those things will feed into these larger efforts. Informally, when we pull this in our trials program, there's probably about 20% of our patients that are just simply self-directed, arriving at us without any sort of community connection to us. And so that's a very valid avenue.
So I think, the comment was made earlier that most everybody wants to be on a phase 3 trial, late stage trial, but there are many, there are fewer of those, many more early stage trials. Suzanne, can you comment on the ethical considerations for early stage trials, as opposed to one that's sort of at the end of the clinical trial path?
Yeah, I mean, that's a really great question. And it all comes to, again, this sort of informed consent and understanding what the goals of the specific trials are. So Yoshi really talked about, what a phase 1 trial is. And when you think about it, sometimes these are called first in man or first in human studies, meaning that we've done some great preclinical work. We think this drug is going to get in, it's going to do what we think it's going to do. We have toxicity studies that we've done in animals and large animals too, which is sort of almost mandated to make sure before we take it into the patient. But ultimately, those early phase studies, sometimes we don't know yet what kind of effect it's going to have on patients. We kind of anticipate it based on the animal models. And so it's important for patients to understand that the goal of like a phase 1 study is such that we don't know yet if it's going to be effective. You're really partnering with us to really explore the very, very early aspect of whether a drug may get in, whether a drug is working at all. And so the benefit there for you as a patient, you need to understand, we don't know yet if it's going to work. And it is incredible to me when I tell patients this, how altruistic they can be and how amazing they are to say, you know what, this may not help me, but if it's going to help the field, it's going to help other patients in the future, yes, I still want to participate. And that to me has always been an incredible piece of the partnership that we have with our patients. And we would never be able to move on to the phase 2 and the phase 3s unless the patients are so courageous to say, absolutely, I will be part of it. I mean, I think Nada could speak to this with the sonodynamic therapy, for example, and some of the work that they're doing where, again, we don't know if this is really going to benefit the patient. And so the ethical considerations there is being honest and transparent with the patients and their families as to what do we really hope to get out of this study. Obviously, we're testing this because we think it's going to make a difference on the tumor biology or, control the tumor better. But that's why we're doing the study. So I think that is one of the most important things about this.
So I'm going to switch gears and take on a different challenge here, one that came up in a question from one of the attendees. And it really gets back to the regulatory question and access to new therapies outside of a clinical trial. So there's many different ways that this has been proposed. Right to try is one term that's been used. Promising Pathway Act is another. What are the challenges with these sorts of ideas? Should we be embracing them, or do they create challenges that are ultimately going to have long-term impact on our ability to develop therapeutics? Let's see, I'll throw that one at Nader first.
So the question, Mike, is it in regards to the passage of new laws, or?
Well, there have been proposals to have right to try. So anything that's been developed in clinical trial, you get to prescribe it and give it to your patient, which is also in a sense like a Promising Pathway Act, where you have a time limit period in which to provide what would otherwise be an experimental therapy.
Right. So so my understanding of the Promising Pathway Act is that it provides a separate sort of avenue for kind of rolling sort of approvals of early stage agents. I don't know exactly how it lowers the bar with respect to those agents getting in, but I think in principle, everyone believes that new agents that are sufficiently safe should be deployed expeditiously to a patient population like ours, where there are few established solutions. So I think that we're already seeing evidence of that more and more with more early stage studies being done in our space. Can we improve upon that? If you ask any drug developer or company, and I think Dr. Heimberger's talk really hammered this home, what are the challenges to getting drugs into patients? Money is one of them, but certainly it's the regulatory timeline and the real massive amount of paperwork that is required. Some of that is essential to establish safety and a track record. Some of that, maybe could be reduced. So I think, reducing regulatory red tape is a great concept. hopefully some of these acts like the Promising Pathway Act does that and doesn't muddy the waters.
Amy, you want to add to that?
I was thinking about how far to go on this response. I would almost, I don't agree with this position, okay, first and foremost, but I will share with you a pharmaceutical view of this from their perspective, which is, let's say you've generated, let's say even a fairly expensive product, let's say a CAR T-cell, you might have a limited inventory on your shelf. Maybe you've synthesized a sufficient amount for, let's say, your 100 patients on clinical trial. And if you have a high demand for these sort of extra patients, and let's say your product is very expensive, then how are you going to cover the cost of that? The other thing is that pharma thinks about in this scenario is the fact that, let's say you do provide this as a compassionate or whatever, except in a sort of non-sort of thought through indication. And the patient has, let's say, profound side effects or dies on the clinical trial. That can shut down your main program. And I know folks are going to be uneasy with me saying this, but that can shut down your main indication in your main program. And I will reference things like gene therapy programs where and maybe they should be shut down. I'm not going to argue that. It's just a fact that you're increasing your liability for something maybe you didn't necessarily develop a drug for in that particular indication. And let's say you're working with VC and you're trying to pull down, 15, 20 million, to run your clinical trial in this indication. And then you have one bad outcome that can shut the whole program down. I'm just giving you the perspective from a pharmaceutical company, not that I personally think that that's the right or wrong thing to do.
So I can give you another concrete example. I think that illustrates some of the challenges, because we're here talking about clinical trials. We do clinical trials to effectively try to prove that a therapy is effective for a specific disease. So as we all know, one of the techniques that we have in neurosurgery radiation oncology is something called radiosurgery. It's a focused form of radiation. And we know that radiosurgery can be very effective for certain types of tumors, particularly brain metastasis. But when it comes to something like GBM, there's actually one randomized trial that showed it's not effective for GBM. Now, there's no one out there marketing radiosurgery for a specific disease because they don't have to, okay? So it's different from a drug. But the fact that we can do radiosurgery for GBM, we don't need to do it in a clinical trial. We certainly can do that as treating physicians. Means that we will never be able to do a clinical trial where we randomize radiosurgery against not doing radiosurgery because nobody's going to go on that trial. They'd rather just make the choice themselves. And we've seen lots of trials where we tried to compare two available therapies or one available therapy with some other type of treatment, and they fail to accrue because there's not sort of a balance, that people don't believe equally that one might be better than the other. They have strong opinions about one versus the other. So that, in a way, that could shut down progress. I think I'm kind of concerned about that. Suzanne, you've had a lot of experience developing therapeutics. I'd love to hear your thoughts.
Yeah, I think the situation there is a little bit different because, it's sort of a available, right, technique. It's same as surgery. What's the role of surgery? How do we show that surgery actually makes a difference? And so it's a little bit different when it's an experimental agent that hasn't been tested before and is now coming to market. So there are differences, I think, in what's available. I think the closest one from a medical point of view is what Yoshi mentioned and what we call off-label use of drugs, right? Mm-hmm. So off-label use of drugs means that those drugs are FDA approved, but we're not using it for which the FDA has given us the label to use it for. And there's some chemotherapies, for example, that are approved and they're used for other cancers, but should it be used for patients with brain tumors? And, I think that I can tell you the one that everybody comes to mind is Avastin or bevacizumab. So it was used for colon cancer. It was shown to be, it has an effect on blood vessels. And everybody thought, well, brain tumor patients, these tumors are very vascular. They have a lot of blood vessels. Let's try it in brain tumor patients. And that's kind of how it started out, right? It was a sort of a example that was done anecdotally, and then there was some effect, and then it became a clinical trial. But I think the prospective evaluation of agents to really demonstrate benefit is really what we want to be at, and to minimize harm. So you've heard from everybody here that just because there's a treatment that's available that might show an effect in another disease, for example, how safe is it in a patient with a brain tumor? Patients with brain tumors, they're on seizures, they may be on steroids, they may be on other medications. That could affect the interaction of those medications with the new drug. So unless we test it to make sure it's safe and then show that it's effective, the sort of anecdotal use of these things can be harmful. So,
Larry, from a patient perspective, any other barriers that you see? Anything else that we should be addressing?
Oh, I think you've covered a lot. I guess I just tip my hat to people who, like the doctors, who go to school for 20 years, practice, and their reward is extending somebody's life 3 months. That is amazing.
Well, appreciate that. Really, really do. One of the areas of great attention now in medicine is something called burnout, because I think that a lot of this is as much as we're here to talk about everything we're doing to try to help brain tumor patients live longer, there actually is a toll on some of the caregivers, both the professional ones and the family members, and these are areas that we're all trying to attend to ourselves, and I know the ABTA is trying to attend to that from a caregiver standpoint, and that's part of why we're doing this today. It's very important because it does take a big toll. Yes, we want to see much better than 3 months.
Yeah,
additional survival.
Having listened to all of this, and God, I know how complicated it is, but sometimes when you're in a binary situation, do something. And I don't know how much the patients can impact that. I hope a lot, but when you've got seems like zero shot, any shot for me, maybe it's my personality, but I would like to see more risk taken, I guess. But I know that's a very complicated subject.
So, Ambreen, do you want to come on and start relaying some of the questions to the panel members? I think we have a little bit of time for that now.
Yes, thank you all so much for this wonderful and dynamic conversation on barriers when it comes to clinical trials. As a reminder for our audience, if you do have questions that you want to ask any of our panelists, please type and submit it using the Q&A button on the bottom of the screen, and we'll answer questions as time allows. So one of the questions we have is asking, when it comes to barriers, how has the COVID-19 pandemic impacted clinical trial enrollment or patients' ability to take part in clinical trials? Dr. Vogelbaum, I'll direct the questions to you, and if you don't mind, maybe directing them to the appropriate panelist member.
Yeah, so Yoshi, we haven't heard from you for a while, so perhaps you can talk about how the pandemic has impacted on clinical trial accrual at University of Michigan.
Sure. So I think in so many different ways, it has impacted things. So, I have a phase 01 clinical trial that my collaborator and I started writing in 2019. And our concept was approved and submitted to IRB in February 2020. And so that was quite a bit, I lived through that in terms of my first big study to go through the IRB. And what I can tell you is it impacted a lot, not just regulatory, but our, ethically speaking, we thought, is it okay to do this when there are pandemic going on? There are not just clinical trial, the pandemic has affected standard of care as well. The neuro-oncology community published a paper about how much temozolomide can affect someone's risk to pandemic and what kind of disease and things like that. So in addition to that, our regulatory people, some of them got COVID. One of my main regulatory person was in ICU for a couple of months. So these things do affect. And in addition to that, we really have to weigh the risk benefit when there are resources or risk, like all of these do affect things. I do think, speaking about burnout, I think things that help me from burnout is not just my hobbies, but also the close relationship I have with my patients, but also my collaboration with people that are here in the panels and also in the community, just knowing that there are people who care to kind of bust through all these barriers no matter what. Because even despite the COVID pandemic, the collaborative group studies kept on happening. The meetings kept on happening. So the COVID pandemic did impact the studies and the speed, and some studies had to be closed if they, surgeries were closed in standard of care. So of course, some of the studies were closed. But things that really mattered, for example, at University of Michigan, there was a algorithm that they used that we all had to submit a questionnaire in terms of if subject in the study population would have any other option if the study would be closed. And if the answer was no, they kept open. So I think most institutions did have some sort of risk benefit analysis of closing the study, what that would mean to patients. So that's how we handled it.
Anyone else have, was it all similar at your institutions, or Suzanne, looks like you want to add to that?
Well, I think if it was something that was therapeutic, that definitely those were maintained for the patients. So that was a good thing. So there was some prioritization. I think it was very difficult though for things that needed frequent visits. So we had to really look carefully at that and talk with the companies about, do they really need to come in however often they were supposed to come in to the medical community environment where COVID was pretty frequent. And all of the non-therapeutics, so all of the studies that I talked to you about partnering, those pretty much were shut down. So the imaging studies, the anything to do with surveys that we might have. And I do think though that pivoting to Zoom, so here we are all in Zoom, right? So we're not physically with each other. We're good. There was good and bad about it, I think. Good in that we could still provide access to healthcare for our patients without them being at risk to coming into the medical environment. The connection though of seeing patients and obviously evaluating them and so forth, it's limited, and there was only so much we could do to accommodate that aspect for clinical trial patients. But yeah, I think the effect on the personnel was also very, very difficult, and we're still struggling with getting people back into the workplace too. In terms of, on a regular basis, because the COVID is still happening, right? So just this week, 3 of our assistants are out. So it's been a challenge for sure.
And when we talk about things like the nursing shortage and the like, we've been seeing that with our clinical trial coordinators. Larry, you talked about one of the clinical trial coordinators who was so important to you. We just actually found out yesterday that we're losing a clinical trial coordinator because she's getting a better paid job outside of a medical center. Just a very common issue, and it's been a real challenge since COVID. Also, we had pharmaceutical companies that shut down trials. So ones that we could have kept open, they shut them down because they couldn't effectively monitor what was going on. That's part of that regulatory framework, and so it was very impactful. Fortunately, we're seeing sort of the light at the end of the tunnel again, starting to improve our clinical trial accruals. I hope that's the case for everyone else. Now, but it was a real challenging time for sure.
Thank you all so much for sharing your insight on that. I think we have time for maybe one more question. This is a great one. And I think if we can get a couple of yours insights on this, it'd be fantastic. So what clinical trials or what research are you as clinicians looking forward to or find to be the most promising at the moment?
Well, who should I pick? Who's going to say my trial? So let's see if we can talk about at a higher level. Again, since we've been addressing things like barriers, okay? What is the most promising approach in general that is likely to produce patient benefit? Maybe Nader, start with you. You've talked about some interesting things, but at a higher level, what do you think it's going to take?
Yeah, I mean, we all get this question all the time, right? And my answer to it is always that if we knew what was most promising, everybody would be on it. So I don't think anybody knows. In a broader sense, Mike, I think our kind of assessment of what can work is predicated on what does work. So what works today? Well, we know that surgery, Temodar, radiation, and tumor treating fields all have very, very high levels of evidence in support of them. They're not curative, but they do something meaningful. And what they all have in common is that they're pretty untargeted therapies. These are all treatments in their own form that generally just kind of cause chaos in the brain in a directed way. And that untargeted element of it is what binds them all together. So in a world of precision medicine and targeted therapies, it seems like so far the things that have worked for us have been relatively untargeted. I think this is why immunotherapy in some ways is very appealing to the field and to cancer in general, because it has a broad sort of footprint in terms of its ability to affect change. Now, we haven't seen that kind of impact within brain tumors yet for immunotherapy, but as an example, I don't know that any of us can read the tea leaves and foresee where we're going to have the next success. What I can say is that in general, for a patient, and this is speaking to Suzanne's comments about early versus late phase trials, I think that in a disease like this, where there are few answers, the opportunity lies in things that have been relatively untested. There are drugs that are in development and have been in development for many, many years that continue to be in trials. And there have been hundreds of patients on those drugs. And it doesn't mean they can't work, but it means that after several hundred patients, it's not obviously making a dramatic impact. Meanwhile, there are many other drugs that are in the wings waiting for opportunities that have simply been untested. And I think that's where there's the highest ceiling. So in general, I try to direct our patients towards earlier phase studies. It doesn't have to be at our institution. And in fact, I think that it's important just to go to the study that fits the biology of your tumor.
So a fail fast strategy is what you're alluding to there, is try to get more agents in, give them a crack. They need to show more than just a hint of activity, and then if they don't, move on to the next one. So I think that's what you were suggesting. That's one potential approach. Yoshi, I think you're at the of this panel. You're in the earliest part of your career. So what are you going to be saying? What are you going to be saying 20 years from now when you're doing one of these panels? What worked?
So I often say I'm a realistic optimist. I see giants in neuro-oncology who have tried their entire career, brilliant geniuses. Do I think I'm going to be any different from them, even at my, try my hardest? Am I going to find the next cure? The reality is I probably will not. However, I can keep trying because I'm an optimist. And I do think, so Mike, you mentioned I study dance. I'm not a basic scientist. I don't have a PhD. If I apply for NIH grant by myself, there's no way I'm going to get it because I don't have a basic science part of my particular career. So I do think in the next 20 years, if we were to make a huge difference, it needs to be from collaboration. I think my specialty is finding loopholes and trying to make things happen. There's a lot of great ideas in basic sciences and labs where great ideas just go to die because they just don't know how to implement it. And they can collaborate with someone like me. I can collaborate with genius basic scientists. I can collaborate with brilliant surgeons. I think collaboration is going to be a key. Glioblastoma is extremely smart as cells. They know how to evade things, not just glioblastoma, but many brain tumors. I think there was a comment about how one of the targeted therapy worked, but stopped working. All these tumors know how to grow, how to figure things out, how to collaborate with other cells. And I think one institution, one lab that is funded by one R01, there's no way that one particular institution is going to find the next answer. And I think there needs to be a way, we all collaborate, but I feel like there's so much more we can do. I do think I'm very appreciative of mentorship I have from the community that are outside of my institution. I feel that it's very selfless of many of you to mentor me. But at the same time, I try to pay it forward as well, because I do think new ideas, they need to know how to implement their ideas as well. And I think just network of the people who care about furthering this field, I think that's going to be a huge key, because great ideas doesn't mean it's going to make a difference unless you know what to do with it.
So
Thank you so much for.
I just want to add one more comment. I'm just going to bring it back to Larry's story because to put things in perspective for our audience, and that is that when Suzanne, Nader, Amy, and I were starting our careers, metastatic melanoma to the brain was pretty much equivalent to GBM. It was just a completely deadly disease, and there wasn't really much to offer except for toxic chemotherapy and radiation, and that was about it. Maybe surgery sometimes. And the initial ideas that led to the breakthrough that we've talked about earlier were ones that were kind of early on laughed out of the grant reviews. The experts said there's no way you can get the immune system to treat a cancer. But with persistence and continued science and translational research, eventually it became clear that this was promising, and then it led to some therapies developed in labs, which then were introduced into patients initially, showed better results than expected, and eventually industry picked up on it. And here we are today with Larry, who really was the beneficiary of all that work. And that's what we're doing for GBM. No one could have predicted that Larry's trial and that drug would have been there in 2016. No one could have nailed down that particular year, just like we can't do the same thing with GBM, but we're going to get there because we have to.
Thank you so much for sharing that, Dr. Vogelbaum. We have certainly come a long way, and it's exciting to see what's on the horizon. Unfortunately, that is all the time that we have for questions. I'm going to go ahead and turn it back to Emily, and thank you again for your time.
Thank you.
Yes, thank you so much, Dr. Vogelbaum, and each of our panelists today for this really didactic session and just discussing the challenges and the impact of the development of cures for patients with brain tumors. And Larry, thank you so much for sharing your personal experience, undergoing a clinical trial. Well, this now concludes the sessions of the ABTA Advances in Brain Tumor Research and Clinical Trials Patient and Family Meeting. As a reminder, today's meeting has been recorded and will be sent out later next week along with some helpful resources. We hope each of you gained some new insight and helpful resources from our meeting today. Before we go, we would love if each of you could help us to understand if you have learned something new today by responding to this last poll. So if you were here with us from the very beginning, we did a pre-event poll where we gauged your understanding of research in clinical trials. So in the very beginning, we had 59% indicate that they had a general understanding. So let's see if our understanding has grown from the beginning of the meeting. I'm going to launch the poll now. Responses are still coming in. I'm going to end it. 3, 2, 1. All right, so as we can see here, we have grown in our understanding. We have 40% with excellent understanding, 36% with great understanding, and 24% with general understanding. So that's definitely an increase from the very beginning, and we hope each of you can continue to learn from what we have discussed today by watching the recording and referencing each of the speakers' presentations. We invite each of you to continue to stay connected and share with us your feedback after this event. Each of you should have received an email just moments ago asking you to take a brief survey to evaluate this meeting. This poll was just a little small example of evaluating and gaining your understanding from the meeting. So we hope each of you can take the time to fill out this survey and provide your feedback with us. For everyone who fills out this survey and includes their email address, you will be entered into a raffle for a chance to win a $25 Amazon gift card. The winner will be emailed the gift card post-event. I would like to thank our program planning chair, Dr. Michael Vogelbaum, for all of his time, effort, and leadership in putting together this incredible and well thought out program. I'm going to hand it back over to you to say a few closing remarks.
Well, thanks again, Emily, and I'd like to extend my appreciation to the time and efforts of our panelists, especially Larry Thomason, who provided a very engaging patient perspective. Hopefully, you've learned more about how we're using our scientific tools and approaches to develop new and more effective treatments for brain tumors and why clinical trials are such an important piece of the path to hope. And Emily, thank you again, and also to the ABTA team for producing this program, and to the sponsors who are critical for the ABTA to pursue its mission of supporting the researchers, clinicians, and most importantly, patients and their families. Thank you.
Yes, thank you so much, Dr. Vogelbaum. And I just want to echo your remarks and saying thank you to all of our speakers for their time and insight today, as well as our sponsors for their support of this virtual event. Without the time and effort from each of these individuals and organizations, we would not be able to bring you this quality educational programming that has brought together the entire brain tumor community. And most importantly, I thank each of you for your time in joining us online today and being a part of this community. We hope you have found our virtual meeting to be full of new information, resources, and support. If you are looking for more content just like this, please check out some of our free upcoming events, such as our webinar on getting past the blood-brain barrier in brain tumor treatment that is taking place next month on July 20th. Additionally, our national conference will be taking place in the fall on September 9th and 10th. Registration for both of these events is open and free, so feel free to check out the links here, or you can use your smartphone and scan the QR code. Thank you again for joining us today, and we hope you enjoy the rest of your day. Take care. Bye.